Enjoy the session.
Hello, welcome all to this workshop of simulating cellular behaviors, co-organized by Marta Lloret from EMBL-EBI and myself from BSc. What we wanted to aim with this workshop was to gather people that was working on different techniques and algorithms that simulate different kinds of cellular behaviors, and if better yet, if they used HPC tools. So you will have many talks today in those 3 hours that we have left. And if you feel interested by any of those talks, please raise questions and intervene and collab and just query people. And also, if you're further interested into this, we are co-organizing as well from the Permit COE project, summer school that is called From Pathway Modeling Tools to Cell Level Simulations, in which some of the tools that will be presented today, you can learn about them there, and also other algorithms that will be presented today using other tools, you can also have them there. So this is you have there the QR code, and you can look for the permitcoe.eu website where you can have more information on this. In any ways, we will be showing again this slide in the following hours, quite a couple more times. So, with much further ado, let's welcome our keynote speaker that will be breaking ground for this session. She's Helana Fertig from the Johns Hopkins Kimmel Cancer Center in the USA. So she will be talking today about machine learning and also trying to find out cellular behaviors with new algorithms. So Alana, the floor is yours. Welcome.
Wonderful. And thank you so much for having me. It's an honor to be here today, and talk today about the work that we've been doing, thinking largely about single cell analysis and new spatial molecular technologies. But one of the things that I do want to bring up is, throughout the session today, I think you're going to see a really nice mixture of, sort of the mathematical model and more of the machine learning from high dimensional data. And I think there's a huge opportunity, from this community and also enabled by high throughput computing to really think about how we're going to bring these together. And something I would love to, keep talking with the audience about over the day. So briefly, my disclosures. And so the thing that I think we can all appreciate and has really come and brought computational biology, I think, to the forefront of the cancer research community is the explosion of new technologies where we now have single cell, genetic, genomic, and even spatial molecular technologies across the full multi-omics scale, where the concept is that if you have this multi-omics information, that you can layer it and get multi-resolution information about the system, where you're spanning the molecular, cellular, intracellular features of a tumor as they're spatially laid out. There's an ability to relate between preclinical models and human tumors. And what this enables us to do is have really, for the first time, a bidirectional approach where we're leveraging the tools that we've developed in computational biology along with technologies that are actually grounding us in the state of a tumor in order to figure out new therapeutic targets, design new therapies, and test them both in preclinical biological models, which of course the word model will be a little bit confusing throughout the day, but mice or organoids or other systems that let us test the use of these that aren't the human tumor before we go directly into human patients. And that's sort of the big scale, I think, goal that a lot of us have in terms of precision, or I would argue predictive medicine in terms of where we need to go. So one case where this has really exploded, I think we saw early hints of this in the cancer world in the precision medicine era with targeted therapeutics. And this has really come into a partnership again, where you have technologies of the tumor microenvironment characterization from single cell that are being partnered with mechanistic advances to immunotherapies that enable us to better attack tumors. And so there's sort of this canonical notion that within immunotherapy responsive tumors, that the tumors present antigens on the surface of their cells that can be recognized by T cells. And the idea of most immunotherapies is then to turn these on by activating silenced immune cells and encouraging trafficking, either through checkpoint inhibitors or through vaccines. The challenge is that a number of tumors are immunosuppressive, where they have a large number of immune cells that are blocking this tumor response and keeping the T cells from coming in. So this is sort of the canonical biomarker, is that you have things in terms of T cell composition versus immunosuppressive environments as your dominant biomarker of immunotherapy response and resistance. But still, even if you use this biomarker, it's not all perfect. Not all so-called hot tumors are responsive to immunotherapy, and not all cold tumors are resistant. And so you want to have this notion of, can we go a little bit deeper into the molecular biology of each of these cells to see, are there other components, are there other factors going in that cause one tumor to be responsive and another to be resistant, that could be the source of new therapies independent of what we're seeing as sort of this dogma of T cell versus immunosuppressive landscape. So what's come in also in the challenges, when we're coupling this with single cell technologies, one of the things that we tend to think about single cell technologies as giving us clusters, and we can use those clusters to give cell types. The other thing is that if you look at single cell data, what you can see is this notion that because you're providing whole transcriptome and information, you have signatures within the data of other phenotypes. And these include the cell state, the cell type, where it is in cell cycle, where it is in evolution, where it is spatially. These all leave a footprint on the data they all leave a footprint on the data that that impact the signal. And so the trick is, if we're trying to figure out what do we want to target and what are our therapies that we want to use, we really want to go beyond this notion of just cell types that's in this classic biomarker example that I gave with T cells versus immunosuppressive cells, and more into these other signatures of phenotype and state in order to figure out what's going on in these cells to develop new therapies phenotypically. So computationally, this notion of transcriptional signatures is mirrored perfectly in the mathematics of matrix factorization, where you imagine you're looking for the underlying signatures within the data. And so machine learning techniques for unsupervised learning at large have really grown and exploded in the era of single cell technologies, where we're trying to understand these continuous states. And so our group, led by Genevieve Stein O'Brien, who's now an assistant professor in neuroscience, has really exploded in terms of thinking about how do we use this data through the lens of matrix factorization so that we can get interpretable features that we can go back to these cell types and try to link them back to the biological processes and also therapies. So the idea behind this is, if we go away from our world of, single cell data where we have tens of thousands of genes and hundreds of thousands of cells, and go into this little cartoon example where we just have four genes under two predominant sets of conditions. Maybe one group on the left is your immunotherapy sensitive, and on the right is immunotherapy resistant, and the cells from that. I think this group here can appreciate that mathematically you can write this data in terms of matrix factorization. And what this is going to enable you to do is figure out, okay, which of the samples are in one state versus another through continuous features, and what genes are then associated with each of those. So on the right, the feature weights that you get across the samples are going to be your continuous cell states that you can map. And on the left, the genes associated with each of these are going to be what are the molecular processes. And so we think about the states as the cell level phenotypes in low dimension as giving you what's going on, and the genes is what are your targets and what's the underlying biology there. So then the trick becomes, how do you do this, right? Because there's tons of different matrix factorization, there's tons of different unsupervised learning methods. Where we come at this from is using a Bayesian non-negative matrix factorization that I developed years ago during my postdoc, and has oddly scaled very well and anticipated single cell data, because what this model had was it had a model of adaptive sparsity where we model each matrix element according to a gamma prior, where the shape parameter is modeled according to a Poisson. And what this enables us to do is it has sort of an adaptive sparsity where if this Poisson distribution is tending to be a large value as it's sampling, your distribution is going to scale away from sparsity and model genes that can be highly expressed. and as it gets smaller, it's going to tend towards more of an exponential and actually be very constrained towards a sparse system. So Genevieve realized that this was very well suited to the dropout of single cell data and the type of sparsity that was there in order to better model the data. And this is one form of ability to find patterns within the data set so that we can start to model these cell states and gene groups. So we had applied this in the context of developmental biology, where the system is much more constrained. And we hypothesized, given the suitability of this technique for single cell data, that it would be very well suited to the understanding the tumor microenvironment. So Emily Davis Marsasaka, a former PhD student in my lab, what she wanted to do was set out and say, okay, there's this data sets and there's a number of data sets with different immunotherapies that we can see, for example, in preclinical sarcoma models and mouse models of cancer. And so what she did is, in this original paper by Gubin et al., what they did is they looked at different immunotherapy combinations and performed single cell data collection and used that for more of the canonical clustering analysis. And so your standard clustering analysis finds different cell types, and they use this in order to associate immunosuppressive cells with different responses to immunotherapy in this original paper. Emily wanted to see if there was something deeper going on in cell states. So what she did was apply our COGAPS approach to these data, and we found several different patterns associated with them. One in particular, we noticed delineated heterogeneity in the NK cells unexpectedly. When we looked at that pattern that was associated with NK cell heterogeneity, what we observed was that it was enriched uniquely in the mice that were treated with anti-CTLA-4 relative to the other groups. So we wanted to see what's going on here. Is this something related to the mechanism of action, specifically of anti-CTLA-4 in this mouse model? So Emily went on to relate this pattern that we learned to pseudotime, as well as to the genes that were associated with it. And doing this, she found that this pattern is identifying markers of NK cell activation during anti-CTLA-4 treatment in this mouse model. So this is really useful in terms of starting to explore what are the patterns in this one data set and what are the features associated with therapeutic response. But the question is then, is this a hat trick of this one mouse model? Is this a hat trick of the mouse immune system? Is this something that's remotely relevant to human biology? So one of the things that we've tried to do is to think about, are there ways computationally that we can start to look between what we learn in these mouse models of biological systems and what's going on in the human tumor? Because ideally, we would like to sort of query between them so we can have this ability excuse me, to do bidirectional experimentation where we can leverage human data sets to query features that we've discovered from the mouse and then go back for mechanistic validation. So what we need computationally is a tool that enables us in silico to query what we've learned between species in a multi-omics manner. So Genevieve and Gaurav had used this notion of, let's imagine we have cogaps as the patterns through which we view the data, this matrix factorization. And we treat each of these sets of gene weights as the lens through which we view the data. Then if we get a new data set coming in, for example, from human, what we can do is to project that new data set into the lenses through which we've divided the original source data and query where that occurs in the new target data set. And then what we can do is quickly ask how specific features that we've learned from one data set are used in another. And what we found in the context of developmental biology is that if you have low dimensional gene weights that reflect biological meaningful axes from transcriptional data, that Projectar, which is our software for querying these new features through transfer learning, can find them. And what we found is that biological features tend to transfer to new data sets, whereas more technical ones like batch and differences between mouse models don't. So Emily wanted to go on to see whether this pattern that she learned with NK cell activation in the mouse was also preserved in human data. So we took in our paper several data sets, including from bulk tumors, as well as single cell data sets from metastatic melanoma immune cells that were treated with anti-CTLA-4, anti-PD-1, or the combination, very similar to the Gubin et al. Data set that used a mouse model for these responses. And so what we did was to query the NK cell activation pattern across these human data sets using our transfer learning method, and then quantify what are the differences in the projected values that we find in human between non-responders and responders. And to our surprise, but also our enthusiasm, given what we were finding, is that we found that this NK cell pattern was enriched in anti-CTLA-4 responders relative to non-responders, but not the other treatment groups, consistent with this being an enrichment of NK cell activation that's associated with anti-CTLA-4 response. So again, we always want to go back and see what's the mechanism behind this. Is this something that's correlative, or is this because CTLA-4 is actually expressed on NK cells? So Alison Fitzgerald and Lou Wiener's lab went forward and performed QRT-PCR as well as Western blots in order to start looking at, do human NK cell lines as well as human donor NK cells express CTLA-4? And she found that this was indeed the case at both a protein and an RNA level. Oh, sorry about that. Let me see if I can do that. Is that better? Okay, great. Sorry about that. I couldn't figure it out with the screen share open. So this, I think, provides sort of a framework where we can start to think about bidirectional research, where we're able to go between mouse, human cell lines and start to query a system by virtue of the patterns that we discovered de novo from the data, and enable both in silico as well as experimental validation of the systems. So, of course, I think it goes without saying, that in the current era of spatial technologies, we've hit a whole new explosion in understanding the tumor microenvironment, because it's not only what cells are there. Even if we have activated NK cells, as I showed you in the last case, it's going to be completely irrelevant what they're doing if they're segregated from the tumor. And so the relative spatial distribution of cells really impacts the therapeutic response, where we could have more immunosuppressive cells infiltrating the tumor, more activated T cells surrounding it, or more exhausted T cells within it. We can have all these different geometries, and each of these are going to give a different response to immunotherapy and require different targets. So we're fortunate that we've gone into this era where the single cell technologies are starting to give way to spatial molecular technologies at both the protein and RNA level, where we can get increasingly high dimensional spatial molecular resolution to really start to tease out the cellular and molecular interactions, leading to therapeutic response and resistance. So as one example of this, what we wanted to do was start to examine the immunosuppressive landscape under therapeutic combinations with this hypothesis that greater immunotherapy resistance is due to blocking of immunosuppressive cells within the tumor microenvironment, as we talked about. And so Mark and Juan, Mark designed a clinical trial that Mark led the correlative analysis on, with the idea of, could you use cabozantinib as a therapy to rewire the tumor immune microenvironment prior to giving immunotherapy, so that way you could start to look at how if you give these combinations testing this hypothesis, can you start to look at how the immune microenvironment is rewired and enable sensitization to therapy? And in this case, this combination indeed led to major complete pathologic response to the immune checkpoint inhibitor. So this trial was designed in such a way that the treatment was given as a lead-in neoadjuvantly, so before any other therapy, and before surgery. So what that enables you to do is get out surgical biospecimens after the treatments so that you can start to query and evaluate what the immune landscape is between responders and non-responders, using these new spatial molecular technologies to start to query response and resistance. So Juan has really pioneered imaging mass cytometry for immune profiling in our group. So he developed a pan-immune panel in order to look at this. And one of the challenges is that, when you're using spatial proteomics, you need to sort of predefine what markers you're going to use, because you have about 40 markers that you can put on the panel. So here he designed a pan-immune panel to look at these samples. And one of the things that I'd like to point out, which I'm not going to talk about, but I think is highly relevant to the high-performance computing group, is that these analyses require a cell segmentation step in order to interpret the data. And that's really an important area of computing research that's emerging for these technologies that really does require interaction with GPU and other styles of computing. And as these data are super high dimensional in imaging, thinking about what are the ways that we can enhance data transfer to clusters for these types of analyses, I think is critical. So again, when you get spatial proteomics data, the first thing that you get is really these beautiful images. And I would say that this is where most people stop. And so you get these beautiful pictures. And what you can see, if you look at these pictures, is that there's a larger number of green cells with E-cadherin markers associated with the tumor in the non-responders, whereas the responders have a greater number of infiltrating immune cells that are coming in to these tumors, consistent with our mechanism of action. And that's associated with a shrinkage of the overall tumor volume with the decreased amount of green cells coming in. So again, you go through, you segment these images, and we want to go to something a little bit more quantifiable. So what you can do is then use these data to apply your sort of standard clustering analyses to start to group different groups of cells in order to phenotype them. So Juan went through and did a flow cell clustering and and use that to determine the different cell types that were there. And indeed, you can go beyond visual characterization into more quantifiable, where you can start to quantify differences between the cell proportions in the groups and use that to observe a decrease in the hepatocytes between the non-responders and responders and an increase in the immune cells in the responders. The other thing that we observe is that, so this is great because it gives you the composition, but we've lost the spatial information that was the whole advantage of going to spatial molecular technologies in the first place. So what Juan did is to further go on and say, okay, now that I have these cell groups, I can take them, I can look at what their average spatial distance is between the cell groups and use that to define a network of how they're responding versus not. And that's what this tree diagram is showing. And so on the left is the average network that's for non-responders, and what's on the right is the average network for responders. And what we observe is that within the non-responders, this orange group of the immune cells versus the green group of the tumor cells are physically segregated from each other. So not only are you seeing more tumor cells, the ones that are there are rarely adjacent to an immune cell. On the other hand, if you look at the responders, there tends to be groups of immune cells that are co-localized with these malignant hepatocytes. So what you're tending to see is that the immune cells are co-localized with the tumor cells associated with the killing. So not only are you getting more of them, they tend to be adjacent and adjacent to the tumor cells as an indication of successful killing. And I think this ability of how do you quantify globally, but also regionally, identify what are the relevant regions of immune killing, and how do you leverage both the spatial information as well as these molecular states is really, I think, a wide open question in the field, and one that's ripe for new computational methods. So as I alluded to earlier, the challenge with spatial proteomics is that you're limited in the number of markers that you're able to query. And so if you want to get a more unbiased view of what's going on in the data, spatial transcriptomics technologies are a phenomenal complement to this because they give you much higher dimensional information across the region of the slide. So Luciani Kagahara went forward on these same samples to perform spatial transcriptomics on these data. So here we had a more limited panel of samples that were available, where we had 4 of the responders and 6 of the non-responders. And the nice thing about these spatial transcriptomics technologies is that they can give you combined imaging on the H&E as well as the full transcriptomics information. So Luciani went forward and applied sort of the same type of clustering analysis, and we recapitulate the same type of thing that we observed within the spatial proteomics data. That within the responders, we have more immune cells relative to the tumor. The number of tumor cells is shrinking, and we have a greater number of tumor cells with less immune infiltration in the non-responders. What was interesting was when we went to some of the samples and we looked at, for example, immune response as marked through the HLA genes, we observed heterogeneity within the tumor regions where there appeared to be different immune responses with different levels of HLA expression throughout the tumor. So Luciani wanted to go forward and see what was associated with that heterogeneity at the immune level across those tumors. And she applied clustering, and we could indeed see more immune infiltration of immune cells within the region on the right using clustering and seeing CD4 and CD8. But in this region on the left, we're really not able to tease apart what's going on in the tumor region. So not surprisingly, we're going to go back to our trusty old COGAPs to see if we can get better segregation of the features in there and better learning of the biological processes that are associated with that. When we apply this approach to our data, what we observe is that the tumor pattern segregates into two distinct patterns. One that's uniquely associated with the tumor region on the left, and one that spans all the tumor regions across all the lesions throughout the tumor. So leading us to hypothesize that there's intra-tumor heterogeneity and different tumor responses that are associated with that. In addition, we see an immune pattern that's infiltrating more on the right than it is on the left that's reflective of the stroma. So we wanted to understand, are there differences in what's going on in interactions with the immune system in these different tumor features that we've learned that are associated with response and resistance? So a tool from my group went on to further look at this, and what he wanted to do was develop a technique of, can we quantify these regions where we see interactions, and then not only do that, go one step forward and see whether we have changes in the immune microenvironment that are associated with that. So I went on further to look at the data and think about, are there places where, for example, there are genes that are changing specifically due to the cellular interactions? So the idea behind this, he developed a new statistic for this called space markers. And the idea behind this is to look at the gene expression changes overall that are observed within the one region, for example, the tumor region, the gene expression changes that are observed in another region, for example, the immune region, find specifically where they're interacting by applying a kernel-based method, and then saying, where are your gene expression changes larger than they would be from just a linear combination of those two over and above what you would see in expectation? And can that give you regions of interaction and what the molecular changes are associated with it? So a tool applied this statistic to these data where we had, again, these two patterns in the immune cell regions. And what he observed with this is that we observed that there was an association, if we went back to these genes, of fibroblasts blocking the tumor immune interactions on the left side of the tumor, whereas there was successful tumor killing by the immune infiltration on the right side. So the question then becomes, which cell type is actually expressing these changes and impacted by these interactions to go forward and define the next target? So we took this the challenge with spatial transcriptomics, of course, is that you don't get true single cell resolution in your data, like you do in single cell data. So what a tool and Shuming went on to do is say, okay, well, we've learned these different patterns. We can take the same notion of transfer learning that we applied from mouse to human, but now take it from spatial data to single cell data to query specifically which cell types do we observe this change in. So we applied this to match single cell data that was generated from the same exact sample. And what we observe is a subset of the cells in which these interaction regions are also observed, predominantly within these orange cells that are associated with the tumor, leading us to hypothesize that most of these molecular changes from the tumor immune interactions are occurring within the tumor cells rather than the immune cells. So with that, I think I've shown you that cellular composition drives immunotherapy response and resistance. Single-cell multiomics technologies are really well suited to resolve the state of the tumor microenvironment. And the computational techniques that I haven't talked about as much today, but we've used throughout the talk, really are critical in order to resolve this accurately, efficiently, and in a way that's more mechanistic than just going to cell types. HOGAP's matrix factorization is a Bayesian matrix factorization that we've developed as one tool for this to identify mechanistic molecular and cellular changes from single cell and spatial molecular data. But again, this is one of many types of tools that are out there. And interaction statistics enable us to model the tumor immune interactions from spatial transcriptomics. And we think that this is just the tip of the iceberg in terms of modeling these interactions and these rules in order to feed into mechanistic mathematical models in order to gain better understanding of the system. And so with that, that's sort of the biological part of the talk. So I'd like to thank everybody in the group. It's really been a huge team effort. Couldn't have done it without just an amazing lab and amazing collaborators. So thanks to everybody as well as funders. And of course, since this is a conference and today, I'd like to sort of shamelessly give, if this is the type of work that you find that's interesting, we're in the process of building a new Convergence Institute that's focused around this type of multidisciplinary research. We're really taking computational techniques, encouraging people to do methodology development, but that's directly applied to clinical translational research. And we're building a new postdoctoral fellows program in order to enable this cross-disciplinary training, where we're going to have a team science approach of mentorship between PIs in data science, cancer biology, and translational oncology. And so both at a mentor level and also at trainees, where you're going to be embedded in grant challenges with clinical fellows. So if this is interesting, I'd encourage people to apply. And thanks everybody and for your attention, and I look forward to taking questions.
Thank you very much, Helana. I see there. I'm hearing myself. So. Okay, maybe now. So I see there's not very quick. Yes, there is a question. Very good talk, an introduction to the biological system. We're going to talk about models during the workshop. In your opinion, does it make sense to start with simple systems and increase the complexity gradually, or we have to go for the full complexity from the beginning?
It's a great question. I mean, no model is going to work if you try to go for the full complexity in the beginning. I think everybody can appreciate that we really do need to parameterize everything and think about what the most crucial variables are. I think the nice thing about these single cell analyses is that they give us a hint into the lenses of what are the possible features that are most relevant. And that's part of why I love this dimension reduction techniques that we use, is they sort of, filter the system down into the most relevant, low dimensional features that are reflecting the biology. And I think the question is, where is that trade-off and what's the right level of complexity? I think one of the things that's challenging, on the biology side, is that there's a tendency to think about, what's the molecular features, can I go as detailed as possible into that cell subtype and gene that's there? And I think, as we're thinking about models, the question that I would sort of ask people is, are you thinking in terms of, I'm only going to select a subset of them, right? So maybe I'm only going to select, CD4 cells and, their different states and go along the whole trajectory. Or do we think at a different scale level, right? So the other way that we can simplify it is to say, okay, T cells do something. There's some behavior in T cells that we can abstract, and do we go to a coarser grain resolution of the biological system, model that, and then refine. So to me, what I would like to see, and what I think is going to be more biological, is to think about, can you go from big scale to smaller scale in terms of the resolution at which you view the system? So as opposed to adding in different cell types or adding in different features that are as reductionist as they are biologically, can we think about what's the biology that's there? Can we make it a bigger picture? And then can we break it down? I don't know if that makes sense, but it's kind of how I think about it. I know you're muted.
Yes, now. And maybe in that way we can also abstract some of this complexity in order to go further up. Yes. So very good point. Yes. So I think they agree that it's a nice answer. Yes. So yes, I don't see any more answers on the live QA. I just wanted to ask you about how do you feel about integrating this kind of analysis with machine learning? I mean, you said it at the end with mechanistic models, right? I mean, do you see, I mean, which one do you see first, or do you see like a feedback loop between them? How would you do this?
So I have a really, so this is my dream. This has been my dream since entering computational biology. And I cannot say how excited I am that we're now here, that like we can actually be having this conversation, right? I remember talking to my mentor when I entered the field like over a decade ago, and he's like, Alana, you're crazy. We're never going to have the technologies. The models are never going to be good enough. And I really feel like we're right on the verge of that. So I will start by saying that my background is in weather prediction, not computational biology. So, and I was trained there in data assimilation. So I put that lens on everything that I do. And that's where I come from. What? Sorry? I
think there was some audio.
No worries. So in weather prediction, what they found is this notion that you need sort of continuous, your mathematical and mechanistic models are going to deviate away from any complex evolving system. Just, you know, because they're going to go into some attractor state, you know, you're going to have errors in initial conditions and things like that. It's an inevitability of mathematical predictions. And no matter how good the model is, we're always going to be faced with that. And so what was found in weather prediction is that you needed to constantly loop back in and sort of nudge the models towards reality through biological data. And so I would love to see us getting into that world in cancer and in biology. The challenge that we still faced is that we don't have enough temporal data in biological systems to do this, at least in genomics and imaging, and I think in other technologies this might happen sooner. So again, I'm coming from this view of weather prediction, but I would love to see something like this come to biology because, you know, mathematically, you know, being trained as a chaos theorist, it's been proven that this is the way you need to nudge systems to make them forecastable. So I think that that's an underlying truth of what we need to do. So, you know, the question is how frequent, what are the data types that we need, what are the types of prediction, which variables are most sensitive, and how do we actually do that, I think is going to be a huge challenge in our community. The other key difference from weather prediction, and I think this gets into your previous question, is that I would argue we still don't know what the fundamental variables are of biology. You know, we have some sense of cells and genes, but we don't know how they function and how they work. And so the ability to start putting them together, figuring out the mechanistic rules from the data, feeding them into mathematical models and iterate, you know, where does it meet the system, where doesn't it? Again, I really think that's going to be the future of this community. And it's something that I personally am very excited to be working on within my own lab. And we'll see where it goes. It is, you know, I'd argue still a little bit more theoretical. And, you know, that takes a little bit more work to get our biology colleagues on board with us and designing the types of experiments and data sets that we need in order to start doing that. But yeah, I'm very excited for the community. I feel like we're at a cusp.
Great. Yes. So there is one more question in the live QA. Either you can answer it like very fast, or you can just type it, the answer. So it's from Vera Pancaldi, and the question was, since you had the chance to work with multiple spatial omics techniques, which ones do you think is delivering less than what we would expect?
I think it really depends on what you're trying to get out of the system. So the spatial proteomics is great because it's got the true single cell resolution, but the trade-off is that it really is dependent on the panel that you're putting in. And so it's really dependent on what cell type, what you're looking for. The spatial transcriptomics I like because it's more unbiased, but it's not at a true single cell resolution, and that limits some of the inference. So, you know, we've been working with either deconvolution techniques through matrix factorization. We also recently put out a preprint where you could apply a machine learning classifier to the H&E data in order to do better cell type annotation and then layer that on to get more refined deconvolution. So I think that we will get there in spatial transcriptomics in terms of it being more sort of single cell, more higher dimensional, but right now that's certainly a limitation. And I think even at a true single cell level, biologists will argue to death about whether protein or RNA is real, is the real signal, and what their gold standard is. And so,
okay.
I think that's, maybe beauty's in the eye of the beholder, but I don't know, my biology colleagues might shoot me for saying that.
Okay. Well, thank you very much for this wonderful talk, and I invite you to keep on. Yes. So yes, I invite you for the rest of the sessions, and let's move now to the selected talks from the call from abstracts. We will begin with Fabian Fröhlich, if I said that correctly, from Harvard University. So, Fabian, the floor is yours.
And also thank you to the organizers for giving me the opportunity to present some of my research to you today. I'll start out my talk by talking a little bit more about the kind of mechanistic models that I've been working with, what the applications are, and why I think they're particularly suited for those applications. A key feature of mechanistic models is that they allow us to integrate knowledge, biochemical knowledge, from different scales. So for example, if we look at the structural level, we know things about binding domains or post-translational modification sites. The molecular level, we know how these molecules interact with each other. And at the pathway level, we know how these molecular processes are organized into pathways that regulate cellular behavior. What mechanistic models now do is that they allow us to aggregate all of that biochemical knowledge into mathematical terms and really formalize all the biochemical knowledge that we have from these different scales. In terms of mathematics, the way that these models are typically formulated is ordinary differential equations, where the state variables of the model are things like protein concentration. Here the state variables is X in blue, molecular species in the model. Those are things like protein concentrations, including phosphorylation status and protein complexes. And the temporal evolution here defined by the function f depends on unknown parameters such as binding constants at catalytic rates or activation energies, as well as known experimental conditions, which are things like drug concentration, media composition, or what kind of cell line or single cell you're using. Now, in general, these parameters P are unknown, so they have to be calibrated on experimental data, and this yields a set of parameters that can then be used to predict how the system is going to behave under different experimental conditions. And one aspect that I'm particularly interested in is prediction of drug combinations, where the model is trained on data for individual drugs and then predicts the effect of drug combinations. This is particularly relevant in the cancer context, as Alana really nicely explained in the previous talk, that these combination therapies are really thought to be the key to overcome drug resistances. However, in practice, due to the huge space of possible combinations, it's not feasible to experimentally assess all of these combinations, but we need some in silico methods to predict the effect of drug combinations. And to put this all in machine learning terms, I think what mechanistic models are particularly suited for is this out-of-sample prediction. We can actually predict things that were not contained in the training data set. Now, one particular issue that comes with mechanistic models is that the underlying differential equation cannot be solved analytically for the vast majority of all models, which means that we have to employ numerical solvers, and simulating these models can take seconds to minutes, which, if you think about that, you have to typically simulate the model thousands to millions of times in order to carry out calibration, puts really a high computational burden on the calibration of these mechanistic models. We really have to carefully think about how to actually do this. Now, reducing the computational complexity generally requires really complex error-prone implementations. In general, we typically use gradient-based optimization methods. So over the course of multiple years, several colleagues and I have developed the computational pipeline here consisting of the problem specification format PTAB, the simulation engine Amici, and the calibration toolbox PyPastel that allow you to go from experimental data to a calibrated model in as little as 4 lines of code, really massively reducing the burden, the burden on the end user for problem formulation and tweaking of individual parameters. The only two that I want to highlight out of these three is Amici, the simulation engine, where I've been heading this respective software development effort together with Paul Stapor and Daniel Weidel, who are a former PhD student and a current senior postdoc with Jan Hasnauer in Munich and Bonn. And together, we've developed this toolbox Amici, which effectively consists of a C++ backend, which allows us to leverage C-level efficiency for simulation, but also provides users with interfaces in Python, C++, and MATLAB, which on the one hand allows really simple interfacing on desktop machines, but also makes the whole setup ready for HPC environments. In terms of development, we've continuously developed Amici for over 7 years now. We've recently really seen a big increase also in papers coming out of labs that don't actively develop Amici, which you see here in yellow. And overall, well, I'm aware of at least 20 research groups in academia and industry who are now using Amici for their day-to-day activities. So why do we put so much effort into developing this piece of software, and why is that actually a worthwhile endeavor? And to answer that question, I'm going to talk about two applications where I'm showcasing a little bit the benefits of Amici. And in the interest of time, I'm not going to talk much about the biological story or the biological insights that we gain from these stories, but really focus more on the methodological advances that we needed to accomplish in order to succeed in these projects. The first project is a collaboration with Alacris Theranostics, which is a small startup company in Berlin. What they developed is a really massive model of cancer-related signaling pathways, used specifically EGFR, ERBB signaling, RAS, AKT, and MYC signaling. And overall, the model includes the action of 7 different small molecule drugs that target individual kinases within these pathways, which you see here in purple. Overall, the model includes over 1,200 molecular species and over 4,000 parameters. So this is a quite large-scale model, much larger than what you typically encounter in systems biology. Now, our collaboration partners were struggling to calibrate this model on data from the Cancer Cell Line Encyclopedia, which is one of these large pharmacogenomic databases. And why were they struggling? Well, they extrapolated how much time it would require to calibrate this model using state-of-the-art methods back then. And I came up with a number of about 10 years. And this is just for a single iteration. So you would have to repeat that hundreds to thousands of times. So we were looking at a completely intractable problem. However, here using advanced mathematical concepts, specifically a joint sensitivity analysis, as well as sparse linear solvers, and sprinkling a little bit of parallelization on top of that, we achieved an over 200,000-fold reduction in compute time, which put us around 3 minutes, which means we really went from a completely untractable to a tractable problem. And for these adjoint sensitivities, the main thing that we exploited was this fact that we have over 4,000 parameters in this model. Now, the second project I'm going to talk about, we had substantially less parameters, so we had to come up with a different strategy. So here in this model, we looked at EGFR and MAPK signaling, so similar pathways or a subset of the pathways that we saw in the previous project. Over here, even though it's a much smaller biological scale, we really have a detailed description of the oligomerization between RAS, RAF, MEK, and ERK proteins, as well as these small molecule inhibitors, which really gave rise to the huge complexity in this model, where we have over 7,000 molecular species that interact by 100,000 reactions. However, really in contrast to the previous model, we only have about 100 parameters, which means that we couldn't really exploit the adjoint sensitivity analysis to achieve the same level of speedups for this experimental for this project. Instead, I'm going to show you this on the next slide, is that we exploited the structure and the experiments in order to gain a massive speed up for the system as well. And here, in terms of training data, what we used is 230 different experimental conditions, including immunofluorescence, transcriptomics, proteomics, and phosphoproteomics data, all in temporal and dose response to 5 different RAF and 5 different MEK inhibitors. And the key insight here is that in all of the experiments, we use at maximum one of those inhibitors. However, if we now look at the summary of different binding interactions, we see here, for example, in the square here shows that the percentage ranging from 0 to 100 of MEK molecular species in the model that are in complex with BRAF. So what this shows is that there's really a huge model where there's tons of different protein-protein interactions going on, which really gives rise to the combinatorial complexity in molecular species. However, if you look at these small molecule inhibitors, specifically the MEK inhibitor, the RAF inhibitor, and the pan-RAF inhibitor, we see that all of those participate in over 4,000 molecular species. However, in any experiment that we consider in the training data, only 1 out of those 3 will be in the model, which means that if we know that they're not present in the experiment, also all of the respective molecular species that are involved don't really have to be simulated because they will have concentration 0. So what we did then, based on this insight, is we constructed smaller submodels that are really tailored to the specific experimental conditions that we're looking at. And you see that compared to the full model on the very right, these submodels are much, much smaller, both in terms of number of species in the model, as well as number of parameters in the model. If we then compared using the full model here on the very right, compared to having these experiment models tailored to the specific experimental conditions, we see that with this multi-model approach, we also achieve about a 30-fold decrease in compute time, where we again go from a time per iteration of about 1 day to 1 hour, which means we can move from a tractable problem, from an untractable problem to a tractable problem. Now, the last thing before I stop is what I briefly want to mention. I think this is also one big topics of this workshop, is that after multiple years of working with really large-scale models, what I've converged towards in my workflow is typically that I use a workflow manager like SnakeMake, which then uses a really predefined set of packages that I find via Docker and that submit everything via Slurm, which means that really all of the analysis that I carry out is really pressing a button and then editing a little bit of code, and I basically almost don't notice at all that I'm working in an HPC environment. And this, on the one hand, makes these kind of analysis really much easier, but also, of course, it vastly improves the reproducibility of the analysis that I carry out. So in summary, I think one thing that I want to really push is that gradient-based methods really allow the efficient calibration of mechanistic models, specifically if you're using a joint sensitivity analysis. And one additional thing that I showed you today is that, sorry about that, is that the experimental structure can really also be exploited to further boost scalability. And I think if we want to think more carefully about how we can actually deploy these things in HTTP environments, we really have to think about workflow and pipeline tools like SnakeMake, Docker, and Slurm. With this, I will thank all of the collaboration partners, especially my mentors and supervisors, Jan Hasener, Fabian Tyson, and Peter Sorger, for making this research possible. And the last thing I want to mention is that I'm going to start my own lab in the spring of next year at the CRIC in London, and there is very generous funding available for PhD and postdoc positions. So if you're interested or have someone who could be interested, please feel free to reach out. I'm happy to take questions.
Great, thank you very much, Fabian, for this talk. I see that there are many open positions between Helana and you.
Yeah,
maybe we have even more as the time goes this afternoon. But that's awesome. So I don't see any open question on the live Q&A. I just wanted to ask about this heat map that you showed about the ERK signaling pathway. Was this like co-occurrence of this protein, or was it? I mean, do you follow the dynamics as well, not only the activation, but also how they activate?
This was really looking at the static structure of the model. So which proteins interact with which, just to have an overview of what kind of modules are there in the model, because we have two big modules. On the one hand, the ERK signaling, and on the other hand, the EGFR module, where we had lots of interactions between the individual proteins. This is just a summary of the binary interactions. We actually also have really many higher order interactions in the model as well.
Okay, great. And also regarding the workflow and the snake-make part that you were talking. So do you have these building blocks or these tools readily available in snake-make? I mean, can we use it already?
So the Docker file is already available. I think that's a huge thing, because for anyone that's ever developed a Python library with C++ extensions, that's just a mess. And especially if you work with Windows computers, installing all of that it's really tedious, and Docker just takes all of that away. So that's really a great advantage. But the snake-make workflows, they are available in the supplement of some of the publications. But I think that's something that I also want to push forward, to have something like a cookie cutter template that sets up everything and then can easily be accessed by other researchers.
Okay, great. So there is one more question on the live Q&A, but we need to move to Pedro. So if you have some time to read to type it just offline, and we can move on to Pedro Monteiro. So our next presenter is Pedro Monteiro from Universidade de Lisboa, and he will be presenting today tools of Boolean modeling simulation. So Pedro, the floor is yours.
Okay, thank you. I hope you can all see. Okay, so I'm Pedro Monteiro. I'm here to present Epilogue, which is a tool that we developed with Claudine Chauillard, that is now in Marseille, but was in Portugal for 5 or 7 years. And so our background is mainly logical modeling, and so we wanted to scale this to multicellular models. So we are used to see patterns in nature, and some patterns emerge as stripes, other as squares, circles, etc. And they have a mostly regular form. And so this was mainly characterized by Alan Turing in 51 or 2 as Turing patterns. And so this is our main motivation. Especially, there was a collaboration that we had with some biologists at IGC in Portugal in the Drosophila segmentation patterns. And we can see here several cells, each expressing a different gene, and here presented in different colors. And so we can see this as an epithelial tissue where there's a rollover. And so we wanted to like I said, scale this, so the modeling of each of the cells, and then integrate the communication between the cells to define a multicellular model. And so we took this approach of composing the logical models with the cellular automata approach. And there was a nice paper in Nature in 2006 by Gibson and colleagues, where they were counting the number of neighbors each cell had in different species, and depending on the size of the cells and the pressure, the biophysical pressures, they counted if they have 3, 4, etc. neighbors. And so topologically, the best arrangement when they are the cells have a common size and they are more or less with equal pressures for all the sides. It's an hexagon with 6 neighbors. And so we fixed this assumption. And so for us, we have an epithelial grid where each position has a single cell, and it has an hexagonal form, which means that it has 6 neighbors. And the goal so now is to have a framework which integrates the signaling between the cells. So we developed Epilogue, which is a Java tool, and it's available for academic and commercial users here in epiloguetool.org. And it's completely open source, so you can go to GitHub, fork it, and suggest pull requests, etc. And so Epilogue actually relies on GeneSim, which is logical modeling tools for cellular models. So we have a unique cell where we have a model with internal components, meaning that we have components that do not have that have regulators defining their states, and then we have input components, which are components that do not have regulators. So this can represent either external signals that can be modified, or represent environmental cues, or just remain constant throughout the simulation. And GeneSim provides several updaters. So these updaters will define the next state of the cell. And these updaters can either generate multiple or single successors. Here, we are going to focus on the updaters that generate only a single successor. So either deterministic, meaning the synchronous updater or priority classes, or the non-deterministic, where we have some random asynchronous update, either with uniform or non-uniform rates. So in Epilogue, then we scale this to multiple cells, and here we are going to use the input components to integrate the signals from neighboring cells. So for example, here on cell 2, this component will integrate the information from neighboring cells of gene G1. So if it's expressed at a given level, then it will the neighboring signal will be integrated in this component. So we have so we define a model with the classical cellular automata where we have integrating functions for the update rules. Okay, and at the multicellular level, we have a synchronous update of all the cells, and at the cellular level, we also have a synchronous update of all the components. So first, we integrate the integration inputs with the information coming from neighboring cells, and then we update internal components. We have these two steps. So this allows us to have an epithelial grid with only a single successor, which is memory efficient and is reproducible. But this is not enough for some of the models. So synchronous models are not very realistic, either at cell or intercellular level. So some models might need to break this synchrony. And so the classical example is the lateral inhibition model, where at the intercellular level, you need to break the synchrony to reach the stability. So in order to achieve this, we consider the approach of alpha synchrony, which was proposed by Nazim Fates in 2013, where at each iteration, we have a population of cells that for which we are going to select a subset, which is a proportion defined by this alpha parameter. So if this alpha parameter is 1, means that we are selecting 100% of the cells, which will be equivalent to the synchronous update. If it's 0, we are going to select only one cell, a random cell, which will be equivalent to the random asynchronous simulation. Or we have an alpha of 0.4 or other value, which will correspond to 40% or other value of cells called to update. So this will now enable us to break the synchrony, but introduces some randomness in the simulation. So we continue to achieve the reproducibility by storing the random seed for which the simulation was generated. And if you want to generate multiple runs, we just use different random seeds. So in this sense, even though we can break the synchrony and introduce some randomness, we still have a deterministic simulation with a single successor in order to be reproducible and memory efficient. So this is the example of the lateral inhibition, where here is represented just 2 cells. And here both have this green component and the red component. So if the green is present in a cell, it will inhibit the red. And then the red will not be able to signal neighboring green cell, which means that the green will disappear. If the green disappears, you'll no longer be able to repress the red, then red will be expressed. If it's expressed, then we'll signal the neighboring cells for green, etc. And then we have this oscillatory behavior. And if we simulate the grid of this lateral inhibition model with a synchronous update, we have an oscillatory grid which the whole grid is red and the whole grid is green, etc. And so to break this, using the alpha synchrony, we can achieve the normal laterally inhibition expected pattern, where, for example, if I have a neighboring cell which is expressing red, at least one, I will be green, for example, and we have achieved this pattern. Of course, here I was so using a different updater. So Epilog relies on BioLQM, and we extended the priority classes defined on BioLQM, which the classical definition only has synchronous or asynchronous classes. And since we needed the introduction of some randomness, so for each class now we can classify it as synchronous, asynchronous, random, uniform, non-uniform. So if it's non-uniform, we can define the rates for each of the components. Okay. And for Epilog specifically, we have a flag that disables the updaters we have, which generate multiple successors, since we only want one grid instead of having alternative features to be accounted in the simulation. And so in this case, in this example, there is a paper from 2008 from Lucas Sanchez and Claudine Chauia, where there's this modeling of the segment polarity module for the refinement of the segmental borders in the fly embryo. And that model here in this example was reduced just to fit here in the example, but it has the same principle. So instead of having the segment polarity genes of engrailed and wingless, I just have here black external and green external, and which will receive the signals from the neighboring cells. And we have 3 cellular markers that here I'm just calling blue, green, and black. And then we defined a multicellular model, which is a grid of 12 by 12 cells with integration functions that are going to be associated to each cell. So for the green external component, it will be 1, so it will be active if there's at least one contacting cell, so one neighboring cell which is expressing green. And black external will be 1 if there's at least one contacting cell with a high level of black. Okay. So in the initial state, we have these segments, which for now in the initial state, we only have green, black, or nothing. And then we want to refine them. And our expected final state, it will be the appearance of the blue lines, so the blue expressing cells. So if we do a simulation using only the synchronous, we actually will have everything blue. So we'll not achieve the expected pattern. If we do the random uniform, we start to see different expressing cells appear, but we lose all the segments. If we do the random non-uniform, so if we start to tweak with the rates, we start to see that there's some pattern emerging. And when we did the synchronous priority classes, where we give some priority to a specific set of genes, we achieved the desired pattern. So actually, since the synchronous is not a very realistic simulation or updater, so we then tweaked the rates on the random non-uniform, and now we can have grids that are reached which conserve more or less the final state and are more realistic, and then we can do several runs to get like a population of grids with the expected pattern. So in prospects, we are working on proliferation, meaning that now instead of having a cell per position, we can have grid positions that are free, meaning that they are not occupied by a cell. And then we can let the cells divide and populate the grid as they divide. This cell division can be either triggered randomly or through a state pattern. So a cell gets to a state where gets divided, and then the other cells can continue, or controlled by an external scheduler. So here we have this work where we have to compute the best direction for cell positioning, either by pressure or so the amount of cells that are pushing cells. So to define which of the empty grid positions the new cell will occupy. And so this work was already developed, but is still in the beta stage, which is not on the main public version of Epilog. Also, some prospects will be to consider cell death and migration, and the variation of environmental signaling through contributions of other cells. So we are working on applying this to an EMT model for a cancer model, and so where we already published the logical model for a single cell, so for the cellular model, and now we're going to integrate it to have an epithelial grid. And we also are working in the integration of Epilog in the Colomoto notebook, where we get rid of this Java interface, and we can have a Python pipeline where we can have a faster integration or test multiple parameters. So just want to thank our collaborators. So Pedro Varela, that was the PhD student that mainly developed Epilog, and Camila Ramos for her master's thesis, where she experimented several stuff, and Adrien, Fauré, and Celine for useful comments on multicellular models. Okay, thank you.
Okay, great. Thank you, Pedro, for the interesting talk. So I think we already stepped on the break.
Sure.
So if there's any question on the live QA, maybe you can answer it when we come back.
Okay.
Yeah, else. So now we will have this break. Let's meet in 12 minutes. So 3 p.m. Barcelona. Yeah, I don't know where from. Where, I mean, 1 hour less for sure in Lisbon. And so yeah, so let's meet in 10 minutes plus. See you. Bye bye. ¿Puedes proyectar mi pantalla? Ahora. Okay, welcome all back from the break, and now we will have a talk by Daniel Bergman from University of Michigan, talking about agent-based models. So, Daniel, floor should be yours now.
All right. Hello, everybody. Thank you to the organizers for inviting me. My name is Daniel Bergman. I'm a postdoc in my third and final year at the University of Michigan. Here we've been developing a global method for simulating intracellular dynamics, or particularly molecular dynamics, in agent-based models. So as a brief outline of my talk, I'll be talking, first introducing some background about agent-based modeling, the computational expense associated with it, particularly that for molecular dynamics in agent-based models and how that motivates this global method that we developed. And then I'll share with you some of the results from that method, as well as some current directions that we're going in and some hopeful future directions, as well as contextualizing it within the greater community of techniques for simulating agent-based models. So background, agent-based modeling, here we're going to have cells represented as agents in three-dimensional space. So similar to the previous talk, the space is going to be partitioned into voxels. In our particular models here that we use, we do have the restriction that there is one agent per lattice site, but I'll say up front that the global method does not need to have that to be implemented, but we've always done it with that assumption of a text-based model. And so with these agent-based models, you update all your cells forward at discrete time steps of delta t. Delta t can be on the order of minutes or hours, depending on what kind of processes you have going on and what you want to capture. So the way that you do this is you have these rates that are associated with all these processes that are going on. For instance, proliferation and apoptosis, you convert that into a probability. And then for all your cells, you give them a chance to do one of those processes, and then you update them all simultaneously. And as I'm sure most people here can appreciate, these rates can vary due to interactions with the local microenvironment, in particular with molecular agents that are diffusing around in the microenvironment. And so one way to capture that dependence on the local microenvironment is through intracellular signaling. Two common examples of intracellular signaling could be interactions technically outside the cell on the cell surface, where cell surface receptors are reacting with diffusing substrates. You could also feed that or have separately gene regulatory networks, for instance, inside the cell that then churn out output that then goes back up to like the mesoscale in the agent-based model to affect different cellular level rates, such as death rates or proliferation rates or cell states. And so importantly, in agent-based models, we're not as often interested in the protein level interactions and the protein levels in these models. We need to have this feedback up to the cellular state. And a common way to model these intracellular signaling models is through differential equation models. So for example, in the image that I showed on the previous slide, where we had an FGFR3 model of bladder cancer, we would take those cell surface reactions and then convert those into a system of ordinary differential equations that we then need to solve for each and every single individual cell in the model. So to update the agent-based model, we first need to actually update the molecular level dynamics. And so this can incorporate roughly 3 different types of interactions. First of all, we need to get the cells or get the molecules to the microenvironment. So for example, this would be PK dynamics. If you have a drug that's trying to get to the microenvironment, it's possible that the substrates that you're considering are produced by the cells themselves. So this PK part is sometimes optional. You also need to move the molecules around the microenvironment. So that'd be solving a diffusion PDE. And then, as I talked about in the previous couple slides, you then have intracellular signaling that determines how the cells then respond to these molecular level dynamics. And so for these three dynamics, you need to solve them iteratively at small little time steps because they all feed one into the next. And then once you've updated those delta T forward in the model, then it's time to update your cellular level. And when you update the cellular level, I want to highlight this point. We make a coarse graining assumption that at that point in time, the molecular dynamics approximate the molecular dynamics for the previous delta T of that time step. So we make a coarse graining assumption in time as we update our cell fate decisions as they respond to those molecular level details. And then we loop this process, and we loop this over and over and over again. And ideally in an agent-based model, we eventually see some sort of emergent behavior occurring on the order of days or weeks. So now I want to start motivating the global method and then how we implement it. So the first thing that we saw in our model that led us to explore the global method was that these differential equations for intracellular signaling took up the majority of our wall time for these models, particularly as we grew larger, or we're working with larger and larger tumor sizes or more and more cells. We saw that those were starting to dominate our wall time as we grew our tumors. The other observation that we made was that the intracellular signal variability was relatively small. We used the signal to noise ratio to measure this, just the average cell signal divided by the standard deviation of the cell signal across all of the cells. And so in this framework, a high SNR value means small differences between the cells. And so over the course of 8 simulated days with 5 doses of therapy, we saw at the lowest level, the signal noise ratio was about 30. So again, that means that the mean was about 30 times higher than the standard deviation. So because of that, because we saw that there was such low variability between all of our cells, we then decided to use that to build the first part of the global method, which is instead of solving the intracellular dynamics for every single cell like we did in what we call the local method, we instead cluster together, average together all of the cells and solve the intracellular dynamics one time. So before we talked about how we coarse grain in time the molecular dynamics, here in the global method, we are coarse graining in space as well to solve the molecular dynamics. And this reduces the complexity, the time complexity of solving the intracellular dynamics from the order of however many cells you're simulating all the way down to a constant value, depending on how many times you want to group all these cells together. In addition, there's another part of the global method where we simplify the PDE, the diffusion PDE, basically because we are only concerned about averages of the molecular level dynamics. We average across certain regions of the microenvironment to feed into the intracellular dynamics. So we don't solve the PDE at every single, the fine grain resolution of every single lattice site. Instead, we, for instance, in this image here, you can imagine this blood vessel down at the bottom induces a natural geometry in the microenvironment where the concentration of whatever drug is diffusing out of or extravasating out of that blood vessel into the microenvironment starts at a high concentration near the blood vessel and then decays to a low concentration away from the blood vessel. And then this in turn creates a concentration gradient inside the cells or what's accessible to the cells for their intracellular dynamics. So with those two things, we wanted to simulate our models and see how it compared to the local method, the previous way of solving these agent-based models, and then see what kind of speed up we actually got, what kind of benefit we actually get from simulating it this way. So we saw that, first of all, in comparing the two methods, we saw very strong agreement at the more micro scale, looking at molecular level concentrations and the cell signaling. And then at a more macro scale, we looked at the actual tumor sizes that we saw, the number of cells that we're getting, and we saw very strong agreement there as well. In a different model, we looked at spatial dynamics. Here we have a cell lineage model of IL-6 in small cell lung carcinoma. And we see that the stem cells, the progenitor cells, and the terminally differentiated cells, between the two methods, the local and the global method, we're all ending up in the same spatial location. Even when we look at the molecular level details on a very short time scale, we see that we have very good agreement as far as where this inhibitor is in the microenvironment as it diffuses up from that bottom boundary. And then, of course, we want to see what benefit are we actually gaining from this global method. And we saw incredible speed ups in these simulations here in particular, including as we increase the number of cells that we have. As expected, the total amount of time that we're spending in our agent-based models solving intracellular signaling is heading to zero because, again, we have reduced that time complexity down to a constant order. So where are we going right now with the global method? It's still fairly new. The first thing that we're doing is working on right now is trying to extend this to other different phenomena that happen in biological systems. Those two examples that I briefly mentioned before had continuous effects of the molecular levels on the cells, changing rates of proliferation, apoptosis, or symmetric division in the cell lineage model. But what happens if we have discrete changes? So here we looked at what happens if you have oxygen diffusing through the microenvironment and cells can respond to hypoxic environments and enter into a more quiescent state. So this is ongoing work that we're working on, but we've seen very good agreement there. Another consideration is if you have cells interacting with one another, such as tumor cells and CTLs. Now, under the influence of some antibody, such as anti-PD-1 or anti-PD-L1, now you have three agents that need to work together, the two different cells and the diffusing substrate. Can we still see good agreement there? This is very preliminary work that we're just getting started on with that. So now I want to put all this into the context of where the field is currently with agent-based models. As I mentioned, agent-based models are computationally expensive to simulate, and this often leads to us having relatively small models compared to what are actually in biological tumors. An interesting point is about the access to HPCs, and the intracellular signaling is an embarrassingly parallel problem. However, as has been mentioned in this workshop already, with agent-based models, we often want to simulate many times these models. And so having that access to HPCs, I prefer to use that to run multiple simulations at once rather than having a bunch of cores working together to solve all these intracellular models all at one time. And the community has seen this need to speed up agent-based models to work towards a larger scale of cells. So we've seen work with compartment-based models, or population-based models, seen work with trying to use machine learning to help speed up these intracellular dynamics. And we can think of these all as core screening methods similar to the global method. Kind of my main goal, long-term goal with the global method would be to have an adaptive global method, because of course we've taken fairly simple tumor biology or cell biology so far, but that's not even the case. The geometry that's induced in these cases is not going to hold simply across all different models, and for some models they can change throughout a simulation. And so a big question is, can we adaptively learn these regions? So here we have a snapshot of the PhysiCell model, where you can see that there are geometric regions here that can be clearly defined, but they're not simply just horizontal strips. They're more complex than that. So I'd just like to leave you with this one last takeaway. The global method can achieve massive speedups in simulating agent-based models while maintaining accuracy. I'd like to thank my collaborators who have been with me on this project, Dr. Jackson, Dr. Nazari, originally came up with the idea, and then Drs. Pearson and Swice were there for feedback on the biological models that we worked on. So thank you very much. Thank you again to the committee for inviting me, and I'll take any questions or comments.
Yes, it's good to unmute myself. So thank you very much for the talk. It was very interesting. There's always this question now about if coarse graining or I mean how coarse grain can you go without missing detail. And I think the last part of your talk is quite interesting in the sense of what are the boundaries, what are the limits of this global method. So there is a question by Malvina Marcu. What would have been the scenario if starting with a high cellular variability?
Yeah, that's another, the way I interpret that question is, what if you have cells that respond to the microenvironment with different rate parameters? And that's something that we've also thought about. I would put that almost right up there with the adaptive global method as far as interesting next steps. Our thought there is to try and measure that variability that comes from that. And then after we simulate use the averages of the global method to simulate forward, then try and sample from some distribution and impose that on the cells rather than just copying that value onto all of the cells. But yeah, that's work that we haven't done, but something we've thought about.
Yes, that would be great. Yes. Okay. So as we have tight schedule, as you have seen, let's move on to the following presenter. That will be Eduardo Borghiani from University of Liège in Belgium. So,
yes.
Eduardo, the floor is yours.
Okay, thank you. Hello, welcome everybody to my presentation. I'm Eduardo Borgiani, a postdoc at the University of Liège and K11, and today I'm here to present to you my project called the Agent-Based Model to Simulate Immunocell Response During Early Bone Fracture Healing. For this project, I really focus on the immune response that happens at the beginning of the inflammatory stage of bone fracture healing, because I think that it is a really important stage during this process, especially because it's the starter of the process. Therefore, all the next stages that will happen during that will lead to a complete restoration have to cope with the results of this phase. Therefore, the investigation of the inflammatory stage is essential to explore all the treatment options for bone fracture healing. And I don't intend only the biological one, also the mechanical one. This is why I want to develop a multi-scale in silico model to simulate the mechanobiological processes of bone fracture healing at different time and length scales, with a dedicated focus to the early inflammatory stage. This because actually in literature, it's possible to find a lot of papers that mention in silico model for bone fracture healing, but just few of them are focusing on the inflammatory stage, and I want to overcome this gap. To do this, I started to create an in silico model. For the moment, it's a 2D model, which is based on histological images from MOVAT pentachrome. And for example, I was taking images from literature, like this female mouse bone from a 12-week-old sample of a 0.7 millimeter fracture points. And from this image, I was able to reconstruct, as I mentioned before, a two-dimensional one, shape and geometry that simulates the structure of the fracture in the mouse by using the same sizes and the same structure and same material properties for the different zones. But the part that I'm more interested in is the healing region, or this region of interest that is highlighted here. And I wanted you to focus more on this shape because you will see multiple times during this presentation, as it is the region of interest where all the action will happen. And when I mentioned the action, I mentioned that actually I will include the biological and mechanical model. For the biological model, I start by adding the cellular level, which is for the inflammatory stage characterized by different cells of 4 different phenotypes. 3 of them are macrophages, or M0 for inactivated macrophages, pro-inflammatory macrophages as M1, or anti-inflammatory macrophages as M2. And also neutrophils. Those cells, as it is represented by those arrows, can interact between each other and they can go into differentiation, proliferation, or migration. And it's easier to see the results from this agent-based model that I use to simulate those where all the cells identified by different colors for the different phenotypes can migrate, proliferate, and also, if it is possible to see, they change color or they change phenotype according properly to certain conditions. This agent-based model is based on the code that was used in Physicell and adapted properly for my purposes. But of course, the cellular level is not the only one that I'm interested to. Also to include the subcellular level to investigate this phase. And those two levels have to communicate between each other. And the communication is here represented by those additions that connects the cellular level on the left side and the subcellular level on the right side. And actually, it's possible to see that also for the subcellular level, I was using 4 different elements, which are cytokines that are really important during the development of the inflammatory phase. And those cytokines are tumor necrosis factor alpha, interleukin 10, transforming growth factor beta, and interferon gamma. Of course, all these interactions can be regulated by biological models, which are identified by mathematical equations. And all those mathematical equations are therefore regulated by parameters, which have their own values found in literature or by in vitro experiments. And if I include this additional level, the subcellular level, it's possible to see, for example, here I included the dynamics of tumor necrosis factor alpha. It's possible to see that it's possible to create an interaction between the cellular and subcellular level. Here it's possible to appreciate how the cells tend to move properly according to the variation of this cytokine distribution within the healing region. To simulate this, I'm using BioFVM. And at the end, of course, I want also to include another model, the mechanical model, to include also the tissue level, because actually the mechanoregulation is shown that it's pretty important to identify and characterize the cellular level, and vice versa, the distribution of cells and their capacity to produce matrix and other tissue will update the tissue properties within the healing region, therefore changing the mechanical properties all over the region of interest. To simulate this part, I'm planning to use finite element analysis, but as you can see, it's still a work in progress. But once I will have model together, I will generate the one that I call Combini, which is also known as the computational mechanobiological model of bone injury immune response. But how can I calibrate this model? How can I calibrate in particular the biological model? As I mentioned, I'm using an agent-based model to evaluate the distribution in the space and time of the cells. Therefore, I can use immunofluorescence stained images to stain the distribution of macrophages within the healing region from in vivo data obtained from mouse. And for example, it's possible to see that according to different color and different staining, it's possible to identify the distribution of macrophages of the different phenotypes. And of course, my model is also adaptable according to the same color scheme properly to have a good correspondence between what we observe in vivo with the staining and what we obtain from the in silico model. And therefore, it's possible properly to match together the two models and have a good connection between the experimental world and the computational one, properly to support the calibration of this latter one. Let's move now to how I plan to do the in vivo calibration of my model. First phase is to include sensitivity analysis for the model, because as I mentioned before, the model is based on parameter values, but those parameters are really a lot. Like every arrow that I show you in the initial image is regulated by at least 1 or 2 parameters. Therefore, you can imagine that there is a huge variability in those parameters, and I wanted to investigate how much the model is sensible to each one of those parameters. And to do that, I was using the model with the different parameters to evaluate what's the impact of a variation in the value of a parameter on the quantitative output. And if, for example, we want to observe what's the sensitivity of the model to the number of total macrophages, I can use a sensitivity analysis with a specific design of experiment like Taguchi's orthogonal array to change, according to a precise scheme, the values of multiple parameters and observe from the different results that I get what's the influence of each factor of each parameter on the output, which is, for example, in this case, the number of total macrophages. And this value can be identified by a percentage called the percentage of the total sum of squares for each factor, of course. From these results, you can see that actually it's possible also to graph this percentage of total sum of squares. And it's possible to investigate the sensitivity of the model to each one of the multiple parameters you can see on the right. And from the results, it's possible to see that the most influential parameters are the inactivated macrophages proliferation ratio, which is a strong parameter that has a lot of influence from day 1 to day 3. And also other parameters like the recruitment factor, which have a stronger impact on the model in the early day, in the day 1, because of course it's the moment when the macrophages are more recruited, but it's the model parameter through when the inflammatory stage is coming to an end, so at the end of day 3. While in the opposite side, also the doubling ratio of M1 macrophages, which tend to appear later stage, is more influential at day 3, but less influential at day 1. And another parameter that I want to mention is the debris engulfment ratio, which has a negative percentage of total sum square, because actually it has an inverse effect on the number of total macrophages. If we increase this ratio, we will have a decrease on the number of macrophages. But anyway, it's still an impact. But once we have those 4 most influential parameters, we want to apply an algorithm. For example, in this case, I propose to use a genetic algorithm to see how much the impact of those 4 factors is strong and how much we have to vary the value of those parameters properly to have a good comparison with in vivo data. So for this case, I'm using an in silico data quantitative output, like the number of macrophages, as I mentioned before, and the same quantitative output from an in vivo immuno. And by easily doing a subtraction of the absolute value, I obtain a value which is a score, which I tend to minimize properly because I want to calibrate my model according to reduce the difference with experiments. And to do this calibration, I'm using, I start from the 4 parameters I mentioned before with the values that I found in literature, and I create a range of plus and minus 50% to investigate which are the best values that within those within this range can reduce this score. And as it is a genetic algorithm that is an evolutionary algorithm that tends to find the best outcome and adapt itself, it's possible that actually from a random begin, the algorithm tend to converge to some values which have the most similarity with experimental data. So the least value for this score. In fact, if we do an average score for the model generation after generation, it's possible to see that actually there is a strong decrease, which is already sensitive at generation 4. And moreover, from this huge combination of parameters, it's also possible to evaluate which are the best parameters that can represent the in silico model properly to have a good correspondence with experimental data. For example, here the score is reduced to a number of 2. So there is just a difference of 2 macrophages from what we see experimentally and what my model is producing as an output. And okay, yeah, and here it's possible to see these 2 models, like where only the are displayed, that have a good correspondence in the distribution of in the number of macrophages between experiments and computational side. But As a future work, as I mentioned before, I want also to include the mechanical model, the effect of mechanics on the cellular level, because actually, for example, it was possible to observe from multiple images that actually the distribution of macrophages is not always homogeneous, but they tend to concentrate in a specific zone. Therefore, I want to see that if, by including also a mechanical effect, a mechanical environment, it's possible properly to explain what we observe from in vivo experiment that we cannot obtain from the in silico model with only the representation. I want also to add a postscriptum, because with the support of my dear friend Gabriele, I'm planning to create this Combini environment in Docker, which will include modules that I presented to you until now, like the in silico model, the genetic algorithm, and other modules like for the visualization of the results. And it's going to be generated, of course, in Python, and will let an automatic the possibility to communicate between all the modules automatically. But of course, also this is a work in progress work, of course. And with this slide, I want to thank you all for your attention. And now, if you have any question, I'm here to answer. Thank you.
Okay, thanks, Eduardo. So I think we reached the time to shift speakers. If anyone has, anyone in the audience has any question, please and ask it on the live Q&A, and we will be able to answer it by typing it.
Yeah. So the next speaker is Tobias Dusbel from CERN in Switzerland. Tobias, the floor is yours.
All right, I think we're all set up, right? People can see my face and people can see my slides. Wonderful. So, well, I'm super happy to be part of this workshop today. My name is Tobias Duswald. Yeah. Sure. Wait. What did I just say? All right, wonderful. Okay, so just as I said, I'm super happy to be part of this workshop today. My name is Tobias Duswald. I'm a PhD student at CERN and TUM. I've been working on this project for a bit now. I'm working together with three great researchers, Tom Thorne, Barbara Wohlmuth, and Roman Bauer. They come from Surrey, TUM, and Surrey respectively. And so yeah, what is it about? So the title of my talk is Interfacing the Agent-Based Modeling Software Biodynamo with Approximate Bayesian Computation to Simulate the Growth of Neuronal Morphologies. So there are three important parts here. First, we have Biodynamo to simulate the agent-based forward model. We use approximate Bayesian computation for the inverse problem, and we simulate the growth of neuronal morphologies. And that's kind of like the talk in a nutshell. These are the three core components. Let me briefly go over the agenda before diving into the real part, actually. So I would like to start with giving some biological context, because I'm not sure if everybody is familiar with neurons. I would then like to clearly define the project goal. And from then on, I would like to switch to like a top-down approach. So we will start with the calibration. So what kind of algorithm are we using, and why do we want to use it? And what kind of things does this algorithm require, which is the model, the quantities of interest, or summary statistics, as the algorithm calls it. And obviously data, because if there's no data, then you cannot calibrate anything. And once we have defined those parts, we're gonna take a look at the calibration, what kind of results we get. And it's still a bit of work in progress, so the results are, well, still not all there, and that's what we're gonna discuss in future work. All right, so this slide, I like to call it Neurons 101. I would like you to focus on the top right of the slide, where you can see this schematic picture of a neuron. You see a green circle, that's the nucleus, and it's surrounded by like a purplish bubble that is called the soma. So if I think about neurons, biologists might disagree, but for me, it's like an information processing system. So you have inputs, you have a certain processing unit, and you have outputs. And in terms of the neuron, the inputs are the dendrites, then the information is processed by the soma, and it forwards the information via the axon. And for me, that's kind of like a neuron in a nutshell. What we're interested is in the shape of the dendrites, because the dendrites bring the information to the neuron. And just as for a regular computing system, if the wiring is not working as it's supposed to, you will have certain cognitive deficits. And in that analogon, a computer wouldn't work if it wasn't wired correctly, right? So we are interested in how those dendrites grow and what kind of morphology they show. You can see some examples of those neurons on the lower part of the slide in the black box. I think we can agree that they all look different, but they share some common features. So we're having a kind of like a stochastic model, and we will discuss in detail what we want to do. So let me state the project goal. So what we want to do is we want to have an agent-based mathematical model. We would like to calibrate its parameters such that it generates neurons, and those neurons are supposed to match the largest data source that is available, and that is neuromorpho.org. I would like to emphasize that it's not just some model, it's actually a biological feasible one. So we're not just like, I don't know, throwing out a final state, but we have a growth process that we model, so it's biological feasible. All right, so that's our goal. And how do we get there? We would like to have a calibration procedure. And for a calibration procedure, we need a few things that we need to consider. I guess data is the obvious part, right? We need data, otherwise there's no calibration. So what we would like to use here is approximate Bayesian computation. It's a Bayesian way of solving the inverse problem. If I say inverse problem, I mean we have a model, and given some parameters, the model produces an output, right? Inverse problem does the opposite. So it kind of like figures out the parameters that we have to use to best describe certain data that we provide. Well, on that slide, you see three terms. So first of all, you see sequential Monte Carlo, and you see approximate Bayesian computation. So approximate Bayesian computation is a way to solve the inverse problem in a Bayesian way without considering the likelihood function. So the likelihood can be complicated, and the approximate Bayesian computation dodges that bullet by running simulations of the model in a different way. The sequential Monte Carlo part is kind of like a flavor of the approximate Bayesian computation. So there's many different approximate Bayesian computation algorithms, and sequential Monte Carlo is one of them. It's typically very efficient, and that's why we decided to go for this one. So what does our calibration procedure need? So ultimately, it's the three things on the left-hand side. We need prior parameters. So we do have, I don't know, I think in our model it's like 39 parameters, and for all of these we need a prior distribution. Typically, we just take a uniform one. If we had more knowledge about those, it would be very good to incorporate it in the prior though. Second, we need a model. In our case, we implemented it in BioDynamo, and it takes the parameters and it produces a neuron. And then we need the data, and that's what we're getting from neuromorpho.org. And then in the middle, there's some Bayesian magic happening, and we can compute the posterior, which is sort of the answer of which kind of parameters will we have to choose to explain the data that we provided. A little bit on the HPC aspect of SMC ABC. So this picture is taken from a PhD thesis from Yannick Schelte. I would like to emphasize two things. So basically we have a parameter space and we have a summary statistics space. And I would like to explain how to get from one to the other and what's parallel. So on the left-hand side, you see this big grayish bubble, the dark grayish one. So this could be our prior distribution of parameters. And inside that prior distribution, we choose some particles, so-called particles. It's basically a combination of the parameters that we choose. For each of those particles, we need to evaluate the model. And this model produces, in our case, a neuron. And then on top of this neuron, we compute summary statistics. That could be, for instance, the number of branches, the number of bifurcation points, and so forth. And then we evaluate how close we actually end up to the observed data. But the crucial point is that we have those parallel particles, and for each particle, we need to evaluate the model at least once, usually more often. And therefore, we have a very parallel thing, right? We have independent parameters, and we have to evaluate the model. So this one is very nice use case for HPC, because you can that's trivial parallel. You just run the model for different parameters in parallel. But that's only one aspect of the HPC, right? So we have this model, and this is run multiple times. So what you would like to have is like a parallel outer loop and a very efficient inner loop. And therefore, we decided to implement the model with BioDynamo, which is a agent-based modeling tool that has been proven to be very efficient in the publication that presented it. I think it was a year ago. So this is our forward model. And at this point also, we would like to give some credit to Biodynamic Collaboration, which has been very supportive and does a great job and consists of many different partners. So, but let's go back to the model. So that's what we implemented with Biodynamic, right? So our model in a nutshell produces neurons that look somewhat like that. So we start with the soma in the middle, and then we have these trees that evolve to both sides. The trees are made out of cylindrical agents. You could also call them tubes. I tried to represent that in this slide here. Like if you zoom in, you would see the different agents. They're chained together. It's like a tree-like structure. There's also a continuum in the background. It's stationary, and it guides sort of the basal and the apical tree to one side or the other. But ultimately, we have the cylindrical agents that have fairly simple branching rules. To comment a bit on them, I'll keep it short. We have some rules for branching. We have some certain rules for elongation. At some point we split the agents, because otherwise you just get like one very long one and you cannot really twist it and get the correct shapes. There are some spring mechanics involved because we do not really want the neuron to fold. So they have some internal springs in there to keep the shape. And ultimately we follow a gradient to direct the neuronal growth. So that's our model, but we still need to compute the quantities of interest, because that's our summary statistics in some sense. I don't want to spend too much time, but I would like to emphasize that the summary statistics is really a crucial part of that algorithm, right? Because if the summary statistics are not correct, the optimization or the calibration doesn't get the right information. So these are a bunch of quantities of interest that we would like to consider. Right now, we only consider one. So right now we're working with a very simplified version, but that's our way to get the pipeline stretched out and implemented. And in the near future, we would like to implement all of those, at least the ones that are rather independent of each other. We can compute these quantities of interest on our data set that we generate with the forward model, but also we get it from neuromorpho.org. So neuromorpho.org is the largest data source that is available. And here we get the same morphologies as the ones that we compute. And then we can compute the quantities of interest and compare them. So right now we're using a query. So we consider mouse, neocortex, pyramidal cells, and that all the dendrites must be complete. And we get roughly 1,000 neuronal morphologies. We can then go back to our pipeline. And so we're now coming to the second part of the calibration. We can then drop it into our pipeline and use the SMC ABC algorithm to calibrate our model. We use a Euclidean distance to evaluate the distance in the summary statistics space. We use 50 steps for the sequential Monte Carlo part. We use 500 particles that are largely evaluated in parallel. We right now only consider the mean segment length as the target to calibrate. And we also only calibrate 3 of the 39 parameters. So we're still in a very restricted space. Our priors are all uniform. And at least we can see that the result is clearly not uniform any longer. So we do learn something, but we still need to do a little bit of homework to really evaluate what have we actually learned. I would like to give you some examples of what we actually find. So we have those 3 parameters, and for all of them, I would like to give you some examples in the sense of how does it look on the minimum and the maximum of the uniform prior, and how does the mean look like? So the posterior mean. So if we jump to the basal elongation speed, which is one of the parameters, we only consider the green part right now. We can see that it has a drastic effect on the shape of the neurons, right? And at least the posterior mean is significantly different from the other ones. The same argument holds for basal diameter decrease. Also here we see quite clearly that they differ. And also for the basal branching probability, which has a very intuitive interpretation. The higher the branching probability, the more branches you get. Yes, so these are the results so far. I think we definitely have to do a little bit more work, and I'm really looking forward to doing that and working on the project a bit longer. I think it's very promising. So we need to extend our calibration to more parameters. So right now it's, like I said, only 3, and we have 39 in total. That also yields us to considering other methods, such as possibly active subspace methods to reduce the optimization space. And then we also need to verify our results by seeing how does our experimental data compare to the one that we get from using the posterior. So if we sample the posterior distribution, how do our quantities of interest look in comparison to the data? So that's very high up on our to-do list. And I think that's actually all from my slides. Thank you very much, and I'm happy to take any questions.
Okay. Okay, great. Thank you, Tobias. Looking. Yes. So I think I don't see any questions on the live QA. You said about having different ways of how to optimize these parameters. Can you briefly talk about these future perspectives that you have on this?
Well, ultimately, we want to stick with the approximate Bayesian computation. I think if it comes to different flavors of approximate Bayesian computation, right now the SMC is still our favorite. But what I mentioned and teasered sort of is the part with the active subspaces. So the active subspaces kind of like allow you to find inside the big parameter space, you will like find a linear transformation on a lower dimensional space, which contains the most variability. So it's kind of like you combine like a sensitivity approach together with a with the approximate Bayesian computation, and then you don't have to optimize over many parameters, but well, less. Let's put it like this. So this is something that we think about considering, but we still need to gain a little bit of experience along the way.
Okay. Okay. Thanks. Thank you.
You're welcome.
So we'll move to the next speaker, who is Nina Verstret from INSERM in France. And Nina, the floor is yours.
Hi, can you hear me?
Yes, we can hear you.
I'm going to close this one and here. Okay. Can you still hear me? Oh, moi, j'entends rien. Attendez. Bon, I'll start. So hi, my name is Nina Verstrat. I'm a postdoc in Vera Pancaldi's lab at the Toulouse Cancer Research Center in France. I will present you now an agent-based model of tumor-associated macrophage differentiation in chronic lymphocytic leukemia. So the general aim of this study is to model the tumor microenvironment, which is a complex and dynamic ensemble of cells, including cancer cells, immune cells, and various molecules such as cytokines and growth factors. And well, recent studies have demonstrated that certain features of the tumor microenvironment and its cellular composition in particular play an important role in the outcome of the disease and response to treatment. The macrophages in particular, apart from their phagocytosis activity, have a crucial role on the immune state of the tumor. And well, they are very heterogeneous in their phenotypes, but usually classified as M1 for antitumoral or M2 as for pro-tumoral phenotype. These M2 are also called TAMs for tumor-associated macrophage, as they can promote tumor growth through different processes. Increasing evidence show that macrophages are actually converted to being pro-tumoral by the cancer cells themselves, and we are interested in this phenomenon in this work. So the type of cancer we're working on is called CLL for chronic lymphocytic leukemia. It affects the B cells, it's a blood cancer, and like in some solid tumors, the cancer cells are able to induce the differentiation of the macrophages into TAMs, or nurse-like cells, as they have been named in this disease. So these nurse-like cells basically protect the cancer cells from spontaneous apoptosis and contribute to their chemoresistance. So to study this phenomenon, we propose here an agent-based model of these nurse-like cells polarization that is observed in vitro when we co-culture monocytes and cancer cells. So in our experimental setup, we are able to isolate circulating monocytes and B cells from patients' blood and to cultivate them together in vitro for about 13 days. We can follow the monocytes differentiation into macrophages and further into nurse-like cells, monitoring the cells every 1 or 2 or 3 days to count them and to test their viability by trypan-blue assays. So the hypothesis here is that when the nurse-like cells appear, this provides the cancer cells a protective effect against apoptosis and allow them to survive longer. So in our model, there are basically two main populations, the cancer cells that can be found in their three possible states that correspond to how far they are from dying. So in red, if they're alive but need the anti-apoptotic signals from NLCs. Yellow if they're apoptotic, meaning they are almost dead, or gray if they're dead. The immune cells can also be found under three classes corresponding to their differentiation state. Blue when they are still monocytes, orange when they turn into macrophages, and green for NLCs. So depending on their state, the cells are characterized by specific properties and spatiotemporal behaviors in terms of movement, phagocytosis, or interactions with the other surrounding cells. The model was implemented in a NetLogo software in which time and space are discrete. And basically, the most important behaviors are as follows. At the beginning of the simulation, the monocytes start differentiating into macrophages, which after a sufficient number of physical contacts with the cancer cells will convert into NLCs. And the NLC secrete anti-apoptotic signals, which are captured by the cancer cells, helping them fight death and survive overall longer. Another important behavior is the phagocytosis, is when the myeloid cells are able to eat the dead and apoptotic cancer cells with varying efficiencies. So the model has 19 parameters that are optimized to fit to two different objective functions, the cancer cells viability and the concentration over time, over 13 days that we are acquired experimentally. So about the software's framework that I use, I define all the parameters and rules of the model in the NetLogo model. The parameters which need to be optimized are bound onto an OpenMOL script, pointing as well to the patient's data, and containing also a calibration task that consists of launching 20,000 simulation of the model while exploring the parameters in predefined ranges. This uses the NSGA2 genetic algorithm, which basically evaluates the fitness of each simulation outputs in an iterative manner, modifying the values of each parameter so that the fitness to the objective functions get better over time, basically. So we're actually aiming at a bi-objective optimization of the parameters, and this is the type of results we can obtain after 20,000 simulations, represented here by all these black dots, where each dot is a parameter set whose coordinates correspond to their actual fitness on both objectives. The Pareto front here in red represents an optimal in the parameter search space, in that sense that each set in it is non-dominated, meaning that one objective cannot be improved without lowering the other one. So from that Pareto front, I extract the so-called knee point parameter set, representing an overall optimum in both objectives. And I ran a few simulations of this model version to evaluate how well it would fit to our experimental data set. And this yielded to relatively good scores on both objectives. So the viability dynamics and the concentration dynamics, using both normalized RMSE and R-squared error indicators. So from the optimization results, we can also extract the parameters correlations to both fitnesses, underscoring that there are differential importances of the parameters towards either one or the other objective. And then to further evaluate the performance of this model, I turned out to simulate it with different patients' inputs, namely the initial proportion of monocytes and the initial proportion of apoptotic cells, which are currently almost the only patient-specific data that we possess. So we obtained quite good scores on some patients, while on some other patients, the model fitness would give really worse results, reflecting maybe that performing the parameters optimization on average data might prevent us from understanding the different mechanics or properties that would exist in different patients. So I went back to my data and looking back at the heterogeneity of it, I thought that maybe the features that we're using as inputs are maybe not sufficient to predict the cell's behavior in the context of our model, at least. So I then thought about trying out patient-specific parameter optimization. So basically exploring the model, but trying to fit it to each patient individually, just to see if, if patient-specific models would actually reveal some characteristic values for the model's parameters, or at least some of them. So that's what I did. I ran like 10 patient-specific exploration and chose again the knee point parameter sets for each of them, and ran simulations to evaluate the fit to the corresponding patient data. So this is what I obtained. Overall, looking quite better. And well, yes, expectedly, in general, the error scores were better for the models that were explored in a patient-specific manner compared to the one that was trained on the average data. So that would maybe reflect that the parameters of our model, or at least some of them, might be patient-specific, and that a single set of parameters might not be able to predict every patient's cell's dynamics. So here we can have a closer look at the different patients' parameter sets, and we can see that some parameters take very different values in different patients. So I went on clustering the patients based on these features, revealing that we might actually be dealing with two distinct classes of patients, resulting in very different cancer cell survival dynamics in our in vitro setup. And now, interestingly, we are getting preliminary data with our collaborators showing that these two classes could actually reflect what's been described as protective versus non-protective analyses. And the clustering that we are obtaining through our patient-specific models exploration seems to coincide with what is observed experimentally when following up the NLC formation by FACS. So the differences in the parameters distribution between the two classes is still under investigation right now, but we can already see that some parameters display really different values from one class to the other. And these parameters correspond to properties that might be very well patient-specific, pointing out to some characteristic properties and initialization features in the model. So finally, going back to each patient specifically and having a closer look at their cell viability and concentration dynamics profile, we can indeed observe that for the patients that belong to the non-protective NLC class, their cancer cells show low viability over time, between 60 and 80% after 13 days of co-culture, while the patients of the other class with the more protective NLCs, the viability seems to get rescued to almost 100% at the end of the experiment, therefore showing a higher viability. So interestingly, these two profiles do correspond to what's been described experimentally in other recently published work. And yes, so to conclude, we identified specific sets of parameters allowing us to reflect the dynamics of NLC formation, cancer cells viability and concentration in vitro, both for the average data and in patient-specific manner. We identified a few features that seem to be the most important drivers to fit both our objectives. However, with the patient-specific models, this suggests that these properties, or some of them, might be patient-specific, possibly reflecting protective and non-protective NLC profiles, as they are also observed experimentally. So we are currently investigating how to obtain a more generalized model that could still remain patient-specific and be predictive on the basis of patient features that could be measured before doing the whole co-culture. And in the longer term, we aim at adding other types of cells to better recapitulate the tumor microenvironment, as well as integrating inside each agent their corresponding Boolean gene regulatory models to incorporate molecular signaling pathways. So with this, I thank you, and I thank the members of my team, and I'd be happy to take any questions and suggestions. Thanks. And I'm sorry because I cannot hear anything. I had to close the the bigger screen, so I cannot hear anything.
Okay.
Maybe if I can just read the questions. Otherwise,
Maybe you can write.
I don't know why I can't hear. I had the preview and then I closed it because I had double sound and.
Yes, there is
I
in the
just
main
leave
stage. There
and
is
come
the
back.
little. Yeah, of course. Yes,
Discussion.
at the end.
If two patients' profiles are close to each other, do you also find close parameter sets with the automatic calibration? Yes, I do. Yes. Now I unmuted my tab, but okay, it's fine. I hear everything. So I answered Gael's question. Yes, if the patient's dynamics profile are close, I'm getting similar parameter sets. And that's a good remark because we could also try to cluster the patients based on their dynamics profile. The thing is that we have a lot of missing data, so it's kind of tricky. Then we can simulate each patient and try to cluster with the simulated data. We are getting close results, but not exactly the same.
But I think it was very interesting to see that if you do like a model, you don't get the particularities of all the patients that you want to simulate. And you need to go patient specific in order to get all those specificities, right?
Yes, I'm trying to disentangle a little bit the concept of inputs and parameters of the models and which ones can be fixed and which ones can be varied according to the patients.
Yes. Great. Nice talk.
Thanks.
Thank you very much, Nina. So now we move on to Marco Ruscone from Institut Curie in France. Marco, the floor is yours.
Okay. Can you hear me? Can you see the screen? Perfect. So thank you very much for having me, for allowing me to present you our model. So today I will talk about a multi-scale model of the different modes of cancer cell invasion. Cell invasion is a complex process that can be represented as a series of multiple steps. We started considering a tumor spheroid surrounded by extracellular matrix. And once a cell gets in contact with the extracellular matrix, it will change its phenotype from epithelial to a mesenchymal, gaining more mobility, and it will start infiltrating into the extracellular matrix through the secretion of matrix metalloproteases that cause the degradation of the matrix. So invasion can occur in different modes. A single cell can develop a motile phenotype and migrating by itself, or it can form a cluster with other cells, developing cell junction, developing membrane junction, and migrating collectively. Finally, a cell can start migrating and degrading the extracellular matrix, facilitating the invasion to other mesenchymal cells and forming a stream or a trail. So from literature, collective migration is the more aggressive kind of migration, of invasion, and the objective of this model would be to reproduce the different modes and the mechanism of invasion to do comparison and be validated with other in vitro experiments in order to learn some biological insights. So the software that we used to create this model is PhysiBoss. I will briefly introduce PhysiBoss because Miguel Posteleon in the next talk will talk about it and it will explain in better details. But in short words, PhysiBoss is a multi-scale agent-based modeling frameworks that combine agent-based modeling with stochastic simulation on Boolean model. So basically, each cell is an agent that can interact with other cells, other agents, and also with the substrates presented into the microenvironment. And inside each agent, there is an intracellular signaling network that represents the different interaction between genes and proteins. So in the figure here on the left, you can see the intracellular signaling network that characterize each cancer cell in the model. It is built upon two already published models, one from Cohen and colleagues and the other from Selvaggi and colleagues. It includes genes from the Hippo pathway, ROGTPases, and also the main EMT regulators. And as input nodes for the intracellular network, we have the presence of oxygen, TGF-beta, extracellular matrix, neighboring cells, and DNA damage. Instead, as output, which represents the behavior, the possible behavior for each agent, we have the switch from epithelial to mesenchymal cell growth, extracellular matrix adhesion and degradation, migration, and death for apoptosis. So at the beginning, what we did was to analyze the model. So basically, we studied the possible output of the model with different initial conditions that would represent the agent in different sections of the tumor, like in the center or in the border of the tumor, in order to verify that it would reach the correct phenotypes. So in order to validate the model, we compared the model with some in vitro studies. So here I will present 2 out of 3 studies that we used. The first one is from Helana and colleagues. In their studies, they studied how changes in the extracellular matrix density can affect the different modes of invasion. So in their study, they observed how low non-uniform density induces more single-cell migration. So in order to reproduce this kind of situation, we can control, we have a parameter that control the density of the extracellular matrix in the microenvironment. So we set it as low non-uniform, so low random value. And we, as you can see in the figure, we also obtain the same kind of distribution of single cell. Also in their study, again, they observed that in high uniform density, they induced more collective cell migration. So we proposed the very same thing. We set a high density of extracellular matrix in the microenvironment. And as you can see from the figure, this in our model also induced more collective cell migration. The second study that we used to validate the model is from Moitrier et al. So they observe how local light activation of the Src oncoprotein in an epithelial monolayer promotes collective extrusion, so collective migration. So in their study, they use blue light to activate the Src protein in an epithelial monolayer of cells. And basically, as you can see from the figure on the left, in the blue area, there is a formation of an extrusion of an aggregates that start migrating collectively. So in order to do the very same thing, we changed the initial setting of the model from a spheroid to a monolayer, a disk, basically. And we introduced a new substrate that virtually interact with the cell, introducing and activating mutation of Src in the intracellular model, and this caused also for us the formation of protrusion in the center of our disk. You can notice the Src is also active at the border of the tumor, and this is caused by the contact with with the extracellular matrix. So the model is characterized by a very big number of parameters. So on the left, you can see a partial list of the parameters of the models that are more than 25. Actually, they easily reach 50 between parameters that involve the invasion model and the general parameters of the simulation. And the different modes of the invasion depends on the values of those parameters. We have also to keep into account that each simulation can take between 1 and 2 hours in 3D of computational time. So we wanted to explore how each parameter influences the amount of single and collective migration. So we decided to perform a sensitivity analysis. In order to perform this sensitivity analysis, we had to find a way to quantify the output of the simulation. So we decided to extrapolate the network of the neighbor or for each cell. And as you can see, once we extrapolate the network, we can observe a main component, which is the node in red, single cell that are represented by blue, by the blue node, and in pink we have the cells that are migrating in cluster, and it is possible to verify here also in the image on the right. So as I say, we performed a sensitivity analysis, but on a subset of 7 parameters. So for each parameters, we selected a range and a number of values to try. And for each value, we perform 50 simulation. So for example, in the case of cell ECM repulsion, we perform a total of 500 simulation and so on, so on. So basically, we perform more than 2,000, 3,000 simulation. And in this way, we were able to modulate the presence of single cells or cells in cluster. So we ran the sensitivity analysis on the cluster at Institute Curie, Abacus, which is composed by 28 nodes for a total of 1,120 cores and more than 5 terabytes of RAM. The sensitivity analysis lasted for 2-3 days, if I remember correctly. So it took anyway a long amount of time. And as you can see here on this slide, there are partial results of the sensitivity analysis of 4 out of 7 parameters. So you can notice that despite the high number of simulation that you perform for each value, there is still a high presence of noise. But overall, we can also observe a trend for these 4 values. So for each graph in blue are represented the number of a single cell, and on the side in orange, we have the number of cells in cluster. So both those values in each graph follows a similar trend, but in particular for the parameter migration speed and migration bias, we can observe that for low value of those 2 parameters, we have less single cell and more cells migrating in cluster, as here is for migration speed as for migration bias, while instead for high value of speed and bias, the number of single cell rise and the number of cells in cluster reach sort of plateau. So of course, we had to run this sensitivity analysis on supercomputer to in order not to take an infinite amount of time for us. But we thought to use another method to find an optimal set of parameters. And the idea is to use surrogate models. So basically, we would have a black box that represents a simpler version of the model. And this black box would be trained with the output of our simulation. So then we will feed the black box with a subset of parameter, and after we quantify the output, we would validate the output with actual in vitro experiment, and we see if we have a match. If we don't have a match, then we will feed the simulation with the same output, and in case we will do a retrain of the surrogate model. In this way, we will make sure that we will have a coherence between the main model and the surrogate models. So to conclude my presentation, the model efficiently managed to reproduce the in vitro experiments and also the different kind of invasion. So among the possible application of this model, we could suggest and anticipate potential risk of metastasis, but of course in future release, it would be nice to introduce more genes and pathways also to make the intracellular model more specific, for example. but also it would be nice to introduce more cell types to represent a possible immune response, like using cancer-associated fibroblasts, T cells, or macrophages. And among the limitation is I showing you, we have a large number of parameters, and as the sensitivity analysis showed, we have the presence of lots of noises in the results. And we also have to keep into account the computational time difference between 2D and 3D simulation. For 2D simulation, we are talking about 2-3 minutes. For 3D simulation, 2-3 hours. So I would like to thank my supervisor, the Mabos team, and the Feasibles team for helping us realizing this model. And thanks to the organizer of the workshop for having me. And if you have any questions, I'm really glad to answer them.
That's great. I don't see any questions on the live Q&A, but I had one very fast. In the sensitivity analysis that you do of the parameters, why did you use different values in the ranges? I mean, for some you simulate 7 different parameters. For others you simulate 20 different parameters. I mean, why didn't you just took like 10 and that's it?
Because I started doing the sensitivity analysis using the same number of parameters, and I started with a low number, not a parameter, sorry, of values, and I started with the same number of values for each parameter. But then once I tried to do, once I did the first sensitivity analysis, I noticed that it was difficult to see a trend for some of those parameters, and then I tried to enrich the number of values for those parameters where seeing a trend was more difficult. Instead, for migration bias and migration speed, seeing the trend was easier than, for example, the mesenchymal cell detached threshold. So this is why I'm using different number of values.
Okay. I see a question now. Can you tell us a little more about the data? Is there consistency? We are constantly finding extreme variability in our processes. Do you have the same problem?
Okay, so about the data, I think you mean the data of the sensitivity analysis, I suppose. So we consistency. We managed to find consistency qualitatively speaking, because every time we did a validation with the in vitro experiment, we had to validate, let's say, by hand. But for what is concerning the sensitivity analysis, so the results of how the parameters of the value of the parameters, let's say that we found it difficult to find experiment to compare this kind of data. But it's a work in process still, so we are working on it.
Okay. Great.
So thank you very much.
Thank you, Marco. So we'll move to the last talk of the day, which is from Miguel Ponce de León from the Barcelona Supercomputing Center. Miguel, the floor is yours.
Thank you. Can you hear me?
Yes.
Okay, I'm sharing the screen. I think I should hide this. Can you see the screen properly? Okay. Okay, good afternoon, everybody. My name is Miguel Ponce de León. I'm postdoctoral researcher from the Computational Biology group at the Barcelona Supercomputing Center. I would like to thank the organizer for allowing me presenting my work here. And the title of the presentation is Feasible Solo for Drug Synergy Studies in Real Tumor Simulations. So this is an outline on the presentation, so I will give a very brief background and motivation on biology of cancer and multi-scale modeling, and then how we have been doing on extending the modeling framework, some brief results on simulating and optimizing tumor treatment. So very briefly, so system biology of cancer, it's several modeling challenges. One is cell complexity itself. So cells are very complex systems, and modeling a cell itself is a huge problem. But in addition to that, we know cancer is something more ecological, have several cells that interact with each other and with healthy cells and the immune system, and also we have angiogenesis, and like considering that, it's also another level of challenge. And finally, we have the time complexity with this evolution. So tumors evolve, they are under selective pressure for avoiding, I don't know, immune response or for finding nutrients to grow and invade new tissues and so on. So as we can see, this complex process as tumor evolution span through different time space scales. We are part of an intracellular scale, but then we have processes at the population. So computational multiscale simulation are needed to different levels and how they couple to each other. From the cancer hallmark point of view, there are different modeling approaches or formalism that can be used for tackling different of the processes such as angiogenesis or invasion. So we got kinetic models for simulating the regulated cellular energy or the signaling. We also have population-based modeling for simulating invasion and cell-to-cell interaction and so on. So we had all these different scales and different algorithm approaches for simulating these different processes. But recently, 10 years ago or something, people start integrating these different, what we call hybrid or multiscale model, which basically try to take advantage of the different formalism and merge them together in a huge simulation that has this description at different levels. So in a nutshell, multiscale hybrid models, agent-based, I think it has been presented several times in this workshop, but just briefly, we got agent-based. So agents are represented by cells. There are several ways of representing cells by agent, which is on the overlapping spheres, which is the base of ECCB. Inside cells, we can have different kind of molecular models, signaling, metabolic, cell cycles, and so on. And also we got different time scales, like diffusion operates one time scale, and mechanics of the cells interaction, another one, and cell processing a different, and that's what we call multiscale simulations. So a couple of years ago, Macklin and collaborator, the first release of PCCell, which is an open source physics-based cell simulator for 3D multicellular system, and it's very powerful, it's extensible. So after that, it's triggered the extension of physics by the addition of another layer, which is simulation of signaling or regulatory network, as Marco commented previously. So we start working of PCBOS when I now moved to BSc, and basically the first version of PCBOS lacked some flexibility, so we start recoding it and refactoring the code a little bit to make it more evolvable from the software perspective. We come out with this PCBOS 2, which we have already submitted for publication, and we have completely decoupled the two simulators. Now we have PCCell on one side and simulator, and we create this add-on interface for connecting the two simulators software more clean, and it brings some new features. We have in the microenvironment and the Boolean nodes, as we will see later, how the Boolean nodes can trigger state or changes in the behavior of the cells. And we are in upcoming versions, we will extend the framework to enable mutations on the model, so we can rewire the Boolean model or how it trigger different events on the cell. And at the bottom, you can see an example of the XML configuration. So we can set up the initial condition of the different Boolean model. We can have different models for the different cells and so on. And on the right at the bottom, you can see the links for the repositories. And in develop a very small toolkit called PCTK, which is a Python module for handling physical output and doing plots and analyzing data, which has been somehow useful too. And the other progress we have been doing in the modeling part, it's trying to, or we are on the work on that, but it's creating a distributed parallel or memory distributed parallel version of PCCell. So currently, PhysiCell, the vanilla one, it's distributed in a single computer. You can run using different cores or taking advantage of multi-core architectures of more computers. But if you want to scale up your simulation because you want to increase the size of your tumor, or because you want to increase the complexity of your model, you want to add more intracellular models or both in order to reach realistic simulation scenarios, you will need to taking advantage of parallel structures. So in order to do that, we have been collaborated with Gaurav Saxena for a couple of years, and the first step was to parallelize the partial differential equation solver, which simulate the diffusion, uptake, and secretion of different substrate, which is already published. This is was like the cornerstone of PhysiCell. And then with Goro, we have a parallelized PhysiCell, which is the mechanic solver for the interaction of the cells, and the other part, which are basically embarrassed in parallel. We have a beta version of this. We are testing. It's been more challenging than expected, but we have already been able to run our model that I will present later on a real size simulation, like 10 to the 7 initial cells using 32 nodes or 100 around CPUs. So that's it regarding our contribution on tool development. Now I will present a couple of examples we are using in order to go to or translate these tools into precision medicine. So when first PhysiVos from Gael and now was published, they used this nice example. On the left side, you got a paper on NF kappa beta signaling and cell fate, and they basically describe that under this is experimental data under different concentration and duration of TNF, which is tumor necrosis factor, a cytokine. ELA cells respond in different way. If they are exposed for a longer period of time, become resistant, while pulse is short, then cells go into apoptosis. And using PhysiVos, Gael, Arnau, and Lorenz and the people at QE was able to basically capture some of the experimental result. So here on the plots, you can see the spheroid and how the green line or the green cube, it's the number of alive cells, decrease or increase, or depending on how much TNF, the pulses go very quick or are more separated. We can see different behaviors on the system. So we wanted to pick this example, change a little bit the model with more details, and then see if we are able to optimize these 3 treatment parameters. So basically, we in the new version of PhysiCell, we implement the model with an explicit TNF receptor, and which are described in the paper. And then we use this model exploration framework to try to optimize the treatment. And a treatment is defined by a pulse, an injection duration, TNF. So we would like to find a set of parameters that basically is the tumor to 0, or basically minimize the number of alive cells at the end of the simulation. And for doing that, we use EMUSE, which is a framework stand for Extreme Exploration with Swift, developed as Argonne Laboratory. And basically, it can be used to anything, but it's very popular in agent-based model when you have to optimize very cost black functions. Basically, simulation to assess what happened with the parameter, and what the EMUSE solve, it's a workflow for distributing the simulation, handling the result, communicating the result to some model exploration algorithm, which can be a genetic algorithm or any other heuristic, and then it generates new parameters and distribution and so on. So we explore treatment in different conditions. We optimize treatment in CD, in 2D disk, and in 3D spheroid, and we find feasible parameter distribution. Here we got some examples that non-effective treatment. You can see the green growing to the end, and in the bottom one, we can see that we can able kill all the cells effectively. And the other curve shows this is coarse-grained dynamic of the TNF receptor. That if the receptor is not able to trigger the downstream effect, then you cannot kill cells. You can some cells, but not the majority of them. Whereas in the other case, you just make half of the cells trigger the effect of TNF, and you are able to kill all the cells. There are more examples. You can TNFs, then the cells become resistant. So now that was a proof of to generalize this workflow to enable drug screening in silico experiments. So this is a but this is where everything started. Osmo Slovak from Norway Technical University was working at that time at CENIO with this develop of a Boolean model for predicting drug synergy, a logical model for predicting drug synergy in a line of gastric cancer. So he created this Boolean model. He queued manually all the interaction, and he was able to use the model to predict drug. But at that time, also with Pancaldi, we wanted to add the heterogeneity, which is a key component of cancer resistance, into the model, and that brings us into the agent-based and multi-scale modeling. So currently, we are working on implementing this model. So we are developing this workflow for enabling silico drug screening. So first, to define what's your model. We've seen several models today. You need to do the model calibration. We have finding proper value for the technique. And once you have your model ready and calibrated, you can use it for experiments, these kind of things. So this work is mainly done by Osman Hayun, and we have mimic the experimental setup Osman used when he published. Yes, we have created this plate of cells. We have different drugs. This is a generic mechanism for drug transport into the cell, which binds to a protein, which is one of the inside the Boolean model. So the idea is some kinetic parameter. This is modeled using differential equations, which is played with MABOS, and then transfer functions to transform to continuous to work, and this repercuting the death rates or the best rate of the cells. So the idea is we have a lot of experimental data on the growth on the different drugs and the synergy. So we want to first calibrate the model using the control, then single drugs, and try to predict the synergy between the drugs. And we are using different algorithms for the calibration, ABC, genetic algorithm, covariant matrix adaptation, and so on. The parameters we're working with, diffusion, binding constant, burst, and death rate, and so on. Using different metrics too. We have a root mean square error, or root mean error, or DTUW, which is the also useful. This is just very preliminary results. So we start doing some simulations, and we cells the blue curve on the top left grow exponentially. Realize that we need to add some kind of capacity to the system, so otherwise they will run and keep growing. And we went to documentation of the experiment for running this experiment, and we find that this plateau is due to cell confluence and contact inhibition. So we stopped running the simulation of the synergy, focus on implementing this part. This is what we have. So we implement contact inhibition using pressure-based contact inhibition. And here you can see the red is the average is experimental, and the blue one is the one simulated with physical. So we are happy it's working, and now we are doing the fitting on the other part. This is upcoming work, so this presentation will be continue in the future. So that's mainly it. This is just a summary and a future step. So we have expand the physical simulation framework, adding more intracellular model, also enable the parallel run some applications. So we have this in EMUs to optimize treatments, find feasible regions. Some I haven't discussed it, but different parameter ranges for if you are running in 3D or in 2D, so the space or the distribution is important. We have some on the HPC data, and future or further step are implementing persistent mechanisms. So we have some papers that review common mechanisms, and we would like to implement them so the user can assign probabilities for them to emerge, and finally have this all generalizing workflow to facilitate screening experiment in biology. So that would be it. Thank you very much for your time. And I think we got 30 seconds for. Should I stop sharing?
Yes. Yes.
Let me
So
see.
there are some questions. Tobias asks, I would be interested in how Physibus X distributes the simulation among different cores and nodes, and possibly how they assure load balance with empty volumes that are governed by the PDE but not host any cells.
Okay, I would like interested in how Physibus distributes the simulation among different cores. So the basic it's if you take a bread and you take slices and we have subdomains that we split in different and that's the part that is distributed in different computer nodes. So the domain we split like in to the x axis, and then each node what happens, and it requires communication for the PDE solver. So the PDE, the mechanics are different part of, and they do not to each other. So we can first parallel BioFBM, which is basically, it's not a full parallelization because it lies in one dimension. The solver for solving the PDEs is the Thomas solver, and what Gorov found it can be only partially parallelized. And then we got the mechanic. So the problem is we have been frontier between the domains. So when cells are interacting between and cells one in one domain and domain, where things become a little complicated. But balance, yes, that's a really good question. So if most of the cells are in one subdomain, probably the other subdomain is going to be idle most of the time. So it's we need to work with. So there are other approaches for doing the domain decomposition, like with space filling curves. There is what was the name now?
Piano Hilbert.
Yes, but the software for doing the simulation.
It's called Timothy.
Timothy, right? Yeah, there are more ways of doing that, but also more hard to implement, and also for the bug. So if there's a problem, if you slide along, it's a little a problem. Yeah, it works. So you will find funny things in the frontier of the domain, the arrangement of the cells, but in the big numbers, it reproduces the results we got in a single computer node. Yes, but if you have concentrations of the cell when in the super split, you can find behaviors. Yes, we are working on that to improve a little bit on this.
Then you have two questions on the Q&A. One is from Gael. Do you use the same injection mode as pulses for the two drugs that you combine for synergy, or do you have an optimal injection mode for each drug?
Okay, so we are mimicking Osmond. So in that case, the injection is just you fill the full environment with the concentration of the drug. There is no pulse, such in the case of TNF, that you put a small and then you remove it. You just put. Actually, we are setting the rate of the drug to 0 because we don't know them, and it's an experiment that lasts for 72 hours. So we basically just fill the environment with the experimental data and see what happens.
And then the last one, Jaro, asks you if you're familiar with the Muscle 3 model coupling framework. It is a coupling library to couple your various simulation models together into a multi-scale model. Muscle 3 can be used to control your submodel communication and manage the execution of the models from your laptop to HPC system.
No, I'm not familiar. I'm familiar with some workbench that communicate models, but here PhysiCell it's a standalone C++ coded framework, and one of the nice thing about PhysiCell it has very few dependencies, so it does not rely on other libraries. So the coupling is just an interface written in C++, so we don't need any fancy thing to control. Maybe I'm not in the question completely, but if it does, it's more easy to just write an interface on.
Yeah, I think
Everything
this
is
muscle
C++.
is exactly. I mean, I think this muscle 3 it's useful when you have other scripting languages.
Sure.
Yeah. Okay. Great. Thank you, Miguel, for this talk, and
thank you very much.
all presenters and all attendees that ask questions for their involvement in this workshop. So if I could end with, yes, exactly. So again, be aware that we're hosting a summer school in which you will be able to listen to topics that we talked today and other topics, of course. So please follow this and send us an email if you have any question, and you can contact us with LinkedIn, Twitter, and of course regular mail. So thank you all, and see you around in virtual sessions, and see you around in CGS Barcelona if you're coming face to face. And have a great day and have a great week. See you. Bye bye.